Welcome back. It's day 4 of the Chime 2 workshop, and we have a lot of really exciting stuff to share with you still. Um, I wanted to just very briefly recap what we did yesterday and just kind of make sure that we're all on the same page about what we've done so far, and then give a brief preview of what's coming up here. And then we'll jump right into the first bit of the hands-on activities for the day. So yesterday we covered rarefaction, rarefying, and selecting an even sampling depth. And then we spent a lot of time working through and learning about alpha diversity measures. And then we ended the day with a discussion on beta diversity measures. And so for today, what we're going to do is jump into the portion of computing and visualizing beta diversity. And that will round out the end of the kind of diversity metrics discussion that we have planned for this workshop. After that point, we'll move into some other mechanisms for characterizing our community. So we're going to cover taxonomic classification, assigning taxonomy to our reads, and then we will end the day with discussions on differential abundance and some hands-on computation for the ANCOM method of evaluating differential abundance. So with that, we are going to jump right into that first set of hands-on stuff. That is the Parkinson's mouse beta diversity visualizations, and that video is going to be led by Chris Keefe. And before we get into that video, we are going to play the 4 or 5 minute long connecting to the workshop cluster video again, just so that everyone has some time to log in. Now's the time to get your PDF sheet, make sure that you've got your username and password. And then the video will just roll right into the beta diversity discussion. With that, thank you very much.
Hi everyone, my name is Andrew and I'm going to teach you how to connect to the workshop cluster server using the Chrome Secure Shell app. I'll be teaching you a few other tutorials as we go, so I'll introduce myself now. My name is Andrew Sanchez. I'm a research software engineer at the Pathogen Microbiome Institute, and I'm a new member of the Capareso lab and a QIIME2 developer. We're going to learn about the Secure Shell app, what is it, why we're using it, how to access it from your Chrome web browser, and how to log into the workshop server. So the Secure Shell app is a Google Chrome app, and we're using it so that you don't have to worry about installing QIIME2 on your own computer or on another server. All you have to do is log in to our workshop servers using the Secure Shell app, and you'll have an environment that's ready to go with QIIME2 already installed, so that you can get up and running and start learning right away. The prerequisite for not only this tutorial, but the rest of the tutorials is that you've completed the required software instructions. You go to workshops.qiime2.org and you click the link for this workshop. Scroll down, you'll see the required software section here. Again, you should have already completed this step, and we're going to proceed assuming that you have. Let's get started. So, steps to connect to the workshop server. First, we're going to navigate to Chrome://apps. Chrome://apps. Here you're going to see any apps that you have installed in your browser, and you should see the Secure Shell app here. So, click on that. Next, you need to fill in your username and hostname. Hostname is workshop-server.qiime2.org. Make sure new connection is selected here. Go down here to username. Type in your username and the hostname. You'll notice it's pre-filling in text up here. You don't have to do anything up here. After you type in the workshop server name, all you have to do is press enter. Please note that all this text has nothing to do with your computer. This is actually related to only the workshop server and this Chrome Secure Shell app. Here you'll see a prompt for the password. As you type it in, you will not see any action here. That's for security purposes. It doesn't want to show your password. So don't worry if you don't see anything happening there. Just type in your password and press enter. Again, all of this text here is coming from our workshop servers. So it says system restart required. There's nothing to do about that. Just once you see something like this, you're good to go. This will be different for you depending on what your username is. And congratulations, you've logged in to the workshop cluster server.
Hi everyone, it's Chris again. I hope you're not all as sick of hearing me talk as I am of hearing myself, because we've got some exciting more stuff to talk about. This section is about beta diversity, and here we will be introducing some tools for calculating and visualizing beta diversity measures. If you remember, beta diversity is between sample diversity. So how does the composition of this sample compare to the composition of this sample using some metric of diversity, right? We will be exploring the emperor plots that we built back in the core metrics phylogenetic section. We're going to run the CHIME diversity beta group significance and CHIME diversity Adonis actions, and we are going to use the resulting visualizations to really poke around with the structure of our microbial community's beta diversity, right? I think we're going to learn some cool stuff here, and I think this is going to be kind of a really fun part of the tutorial. So just like in the last section, during which we talked about alpha diversity, we are going to be using data artifacts and visualizations produced by core metrics to produce new visualizations and find new ways to talk about diversity and learn about diversity. And again, just like the alpha diversity section, we are mostly going to be using our visualizations for exploration, trying to find ways of easily visually detecting patterns and things in our data in ways that we can't really do by just looking at numbers. And we will use the statistics generated by these methods to quantify and test our hypothesis in ways that we can talk about and publish. I think we can kind of dive right in. We're going to be starting right out of the gate with questions this time, and that's kind of a fun way to do it. And we can do that because we have already created the visualizations that we're going to be using. So rather than going straight to the terminal, I'm going to head back to the files I've got stored on the workshop server. I'm going to click into Core Metrics Results. You'll recognize this file by now, this directory. And we're going to grab a couple of our Unifrac Emperor plots. You can see that these are QZVs, they're visualizations. And they're both really nicely and easily named, so we shouldn't have too much trouble finding them. Just as before, I'm just copying these URLs, pasting them into QIIME2 view, and you should be able to follow along pretty easily. And also, just like in the alpha diversity section, I'm going to be opening up two tabs with two visualizations side by side. And like I said before, that can be really helpful because it allows us to compare and contrast two different metrics to see what we can learn kind of when we zoom out and look what those metrics themselves mean, right? We can see that starting from the same data, we've produced two remarkably different plots here. I'm gonna start this one by kind of asking the questions And then we'll get into these visualizations. We'll look at what they are and how they work a little bit mechanistically. And then we'll try to answer some questions, right? So first question is, in the unweighted unifrac emperor plot, can you find separation in the data? If so, can you find a metadata factor that reflects the separation? What if we used unifrac distance, or weighted unifrac? So we can kind of dive right into this. When we look at this unweighted unifrac plot, it immediately jumps out to my eye, and I'm sure to yours as well, that we have two clusters of points here, right? And this big gap separating them. So right here along this axis 1, we do very clearly have a separation. What that separation means is kind of why this emperor plot exists. And so I'm going to zoom out a little bit and we'll talk about what we're looking at here. Emperor is a tool that allows us to plot principal coordinates analysis of our data. It takes in a distance matrix, if I'm remembering correct. Well, that's not true. It takes in a PCOA data set. And that PCOA data set is calculated from a distance matrix, which is calculated using unweighted unifrac in this case, or weighted unifrac in the other case, or whatever other beta diversity metric you prefer. The way this plot is designed, it is interactive, so you have the ability to really pan around these three most important axes in your data. These are the axes selected by the model which contribute most heavily to difference in the data. And when we open a new emperor plot, by default it opens in an orientation that best represents or maximizes the amount of spread or separation in the data. So whenever you open this, you kind of start with an optimal picture. And panning around can just give you different views of data in your metadata that can sometimes be useful. So here we are, we're looking at this thing, we found this separation, and we're supposed to find out whether there is a metadata category that describes that separation well. There are a lot of tools for doing that here. You'll see a whole list of them along this menu on the right. And we're going to start with color because color is a really quick and easy way to see patterns in data. If I, for example, color by cage ID, cage 31 is represented by these points in red. Cage 43 is represented by these points in green. This is notably a categorical variable in our data. So using these categorical colorings, like this color palette that is designed for that, is a really reasonable way of looking at this data. If, on the other hand, we were looking at days post-transplant, using one of these sequential colorings might make more sense. In this case, we don't see a clear pattern, but if we had a trend visible over time, you might be able to track the low values moving into high values in a pattern of increasing color gradient in one direction or another, right? So if we imagine, let's pretend we're doing a study on temperature, and we see that diversity increases according to temperature, we might see, as these temperature values increase, a gradient in the scatter plot on the visualization in front of us, right? We're going to step back to these classic QIIME 2 colors because we're not working with continuous data here. And we're going to start looking at some of these other categories to see if we've got anything. We see some clustering with cage, but there's still a lot of mixing. We see, let's look at genotype. Genotype is really important to this study. This is probably where I would start, actually, if I were just doing this this exploratory work on my own. And again, we see some clusters here, but nothing that's really clear cut and matches this big separation. If, on the other hand, we look at donor or donor status, it becomes immediately apparent that all of our healthy mice plot here with one outlier exception, and all of our PD mice, all of our humanized mice, plot over here. So the difference in our data is being explained very well by donor status. What is that outlier? I'm not really sure. It's possible that this is the same outlier that has shown up in other places. If I click on that point, it's possible to get, and you see at the lower left, the sample ID of that outlier point. And so you can go back and look at your metadata and see exactly, which cage that came from, how it was treated, if there are any other unique things about it. You can check your lab notes and figure out whether maybe there was a contamination event with that one sample, or something happened that was unexpected there. And so being able to look into these points lets you look back and just make sure that everything's buttoned up on the experimental side. So we can do really similar things with shape, for example, where we color, or let's say we shape based on genotype. And this allows you to look at more than one of your metadata categories in one plot. You can use size, scale, to do similar things, scaling up certain categories or scaling things according to a data value. Visibility allows you to drop things out. What I'm wrestling with right now, though, is that some of these blue points, they just don't stand out super well to my eye. And especially if I were going to try to make this into a publication figure, I would probably want this oriented with a white background and a colored foreground. And so if we click over on axes, this is where a lot of the controls are for kind of the visualization's components. We can swap the axis and label colors to black and the background color to white. And all of a sudden we've got a slightly more contrasty way of looking at the same visualization. Another really neat tool that we can use here, let's say that we really only want to picture the two most important axes. I can drop out that third axis and look at this in kind of like square 2 dimensions, which makes it really easy to visualize the two most important dimensions. And then, for example, save our image as a vector or raster for publication. Or even if I have strong feelings about how this looks and I'm willing to put in the time to learn a little bit, I can open this image with Vega, which is a kind of a visualization editing tool, and on a very fine-grained level manipulate how each one of these points is represented, and then output the figure that I want. While we're in here playing with the nuts and bolts of this visualization, I'd like to show you guys one more thing. That is this save and load current settings feature. You can, if you remember, when we first jumped into this visualization, it didn't tell us a whole lot, right? I just reloaded the visualization so we can see it as it was originally. But let's say we find something that's really interesting and want to communicate it quickly to a reviewer, a PI, a collaborator. We can save the settings and send those settings to our PI alongside the visualization, and then they can load them and see exactly what we were seeing after configuring this thing in the way that best represents what we see in the data. All right, I think that's a pretty deep dive into emperor plots. There are lots of other things that you can do here. Animations are possible in this tool, which is pretty remarkable. And we'll see those a little bit later when we talk about longitudinal study. But I think it's time to get back to the core of what we're doing, which is trying to understand our data and answer questions about our hypothesis, right? So if we step back to these questions, we've looked at our unweighted unifrac emperor plot. We found a clear separation in the data. We found a metadata factor, donor, that reflects that separation very clearly. What if we looked at weighted unifrac? Tellingly, we also have a clear separation here. It's not as strong. You can see that though that separation exists, it's nowhere near as significant as the separation in unweighted unifrac. What that allows us to do is consider these two different metrics side by side and maybe better understand why our community separates differently in those two different metrics. So if you remember from the lecture the difference between weighted and unweighted unifrac, you'll know that weighted unifrac takes into account the relative frequency of the features represented in the data. And the fact that we're seeing less separation in this weighted plot than we do in the unweighted plot suggests that maybe in the unweighted plot, which arguably overrepresents rare, like infrequent features, some of this difference is coming from all of those smaller feature counts, right? These are the occasional 1, 2, 3, 10 count features in our subsample data. Whereas in the weighted unifrac, we see a little less difference because the most prominent features are present in both communities. And so that is kind of washing out some of that difference that we see in the smaller features. Now, I don't know that that's true, but that seems like a reasonable way to interpret the plot. And it's a thing that we can kind of carry with us as we keep thinking about our data. So another really useful thing that we can use these visualizations for is trying to find patterns that indicate bias, right? So imagine you have done all of the sample collection and sequencing and live animal work that goes into a mouse study. There are a number of places where bias can creep in. It can creep in from co-housing animals in the same cage. It can creep in during sequencing. If you had multiple sequencing runs, differences in how those runs proceeded could produce differences in the way the data actually rendered. And so making sure that we track in our metadata things like cage ID for each mouse and which sequencing run a sample came from, etc. Which extraction group some given sample was in, allows us to look at those things really quickly and see whether we're getting the same kind of clustering or whether we're getting a coloring that describes some separation in the data, right? Here, with cage ID, we do see some clustering. You know, the blues and reds and yellows are kind of all together, and the greens and yellows and purples are all together. But I suspect that there's a lot of mixing of colors, right? And I suspect that that separation is much better explained by donor than it is by cage. You could run some statistics and quantify your cage effect, essentially. And we may find that there is cage effect in this study that is important. But for now, it doesn't look like we have a cage effect problem. Now, as I'm sure you guys all recognize, though we're getting some really amazing insights really quickly just by looking at these emperor plots, these are qualitative insights. I can't tell you, for example, how different these two things are. I just can tell you that there's clearly some separation along axis 1 of this principal coordinate set. So we're going to move on and start getting into trying to quantify the trends that we see here. We're going to do that first with Permanova. Permanova is a hypothesis test, and it tests whether samples within a group are more similar to each other than they are to samples in between groups. And that will let us ask about the significance of our hypothesis, essentially. It'll allow us to test our hypothesis about genotype and its relationship with diversity. I have copied these commands. I have failed to log into the workshop server, so I'm going to do that again real quick. Ooh, not winning any points this time. All right, we're back in business. So we've got our workshop directory. I'll cd into workshop. I'll look around. And let's just walk through these commands real quick. A lot of this is going to look very familiar. These are very simple commands in terms of the inputs that they take. We need a distance matrix, which we will get from core metrics. We need the same metadata TSV that we've been using. We need to choose a column in that metadata that we care about, right? So here we're looking at donor because donor is the covariate that's shown us the strongest trends so far. A little farther down, we'll try another one. We'll look at cage, but you could run the same commands using genotype if you felt that genotype was had a lot of potential as an important factor here, right? Donor shows a stronger signal in this data set, and so that's what we'll start with. But obviously, if this were your study, you would probably look at them all. It looks like I did a weird thing. Somewhere in the process of copy-pasting this, I produced something strange. Core metrics results, weighted unifrac donor significance.qzv squiggle.qzv. And that's probably not a problem. It's probably just a funny file name. Yep, so here we are with .qzv.qzv. I'm going to rerun that command so that we can get rid of that guy. First, I'm going to remove the old one because I don't want extra stuff hanging around. And I just have to remember that I'm still in the workshop directory, and I'm removing something from within a subdirectory. So I have to give the file path, the relative file path, to my weighted unifrac donor significance QZV. And there we go. That's all cleaned up. And I'm going to once again copy this command, hit enter, and we're in business. I will copy the second one. We'll do the same thing. we'll navigate back to the server, refresh, and we've got a weighted unifrac donor significance and an unweighted unifrac donor significance that we can bring up in Q2 view. So here we are looking at our unweighted unifrac significance on the grouped by donor. And what I mean when I say that is we are grouping our samples by donor and then running a permutation test using Permanova on those samples, and specifically on the unweighted unifrac distances of those samples. This summary table at the top tells us essentially about the test that we have run. We know that we use Permanova, that the test statistic is a pseudo F, that our sample size is 47, so there are 47 samples in our test, and that we have two groups, right? We have the control and we have the humanized Parkinson's mice, right? This is where we get into the fun stuff. Our test statistic at 21.24 is pretty high, and our p-value is quite low, and therefore is showing that donor is significantly correlated with difference in the unweighted unifrac metric, right? So that gives us really our answer, right? We have quantified difference here, and we've confirmed that donor does make a difference. And as we come down here, these plots give us a nice visual picture of what those distances look like. Here, we are looking at distances from healthy control samples to healthy control samples, and from Parkinson's mouse samples to healthy control samples. Here, we are looking at the distances from those Parkinson's mice to Parkinson's mice and healthy controls to Parkinson's mice. On our y-axis is our distance. On our x-axis are the groups that we're testing. And the box plots that you see are implemented, I believe, in the same way as the alpha group significance box plots, where you have the 25th percentile of our samples, the 50th, the median, the 75th percentile, and then out here we have our outliers, which are, I think, 150% of the interquartile range. Each one of these dots does represent an outlier sample, and you can see pretty quickly that we do have a sizable difference in the way this data is structured. When we look at weighted unifrac, we have many of the same things happening up here. Still have a significant p-value. There's a difference correlated with donor. We're still running a Permanova test with 999 permutations. But you can see that these plots are maybe a little bit closer together, which reflects pretty well what we saw in our Emperor plots. So before we leave these plots behind, there are a couple of nice features I want to show you. First, they give us the ability to pull down our visualizations as PDFs with a click, right there or right there. And we can also pull down the raw data in a TSV format. I don't think I've spent any time with you guys looking at provenance graphs during these tutorials, so we'll take just a moment to look at this. I'm sure somebody else has already walked you through one of these, but when you're using Q2 View, it's really easy to visualize kind of the process whereby you arrived at the visualization you produced. These graphs move from top to bottom, first steps to last. You have an import process here. And each step of the way, inside the circle, you have the type, format, and unique identifier of the artifact or visualization that was produced. So here you can see that we pulled in our demultiplexed seeks and a reference database. We ran data2, which produced feature data and a feature table, and allowed us to start building a reference phylogeny, calculating our distance matrices, and creating the visualization that we've been looking at here. Which visualization helped us answer a really important question, showing that there is indeed a connection between donor and both weighted and unweighted unifrac beta diversity metrics. You'll notice down in the questions here, the first question is there a significant effective donor? And I think it's pretty safe to say we've checked that off the list as a resounding yes. I am going to next run these commands also using the beta group significance visualizer. And you'll notice before we run them that there's an additional argument this time. We are using p pairwise. And when you think about the data that we're visualizing, up here it's donor, where we have control and experimental groups. Down here it's cage, and we clearly have a much larger number of cages than 2. So using p pairwise allows us to pairwise test individual differences between cages and see how cage differences influence difference. So I pasted these into Secure Shell, hit enter, and you guys know the dance by now, you've done this a few times. Produced the 2 visualizations shown here. We'll flip back over to our browser, refresh. We see we've got some nice new things to play with. An unweighted unifrac cage significance, which we can copy and paste in. And a weighted unifrac cage significance. Again, I'm just right-clicking, copying, and pasting. And most of this visualization will look very familiar to you. You'll notice that the number of groups has jumped from 2 to 6 because we have 6 cages. You'll notice that we still have this uncorrected p-value of 0.001. But as you come down, you'll notice that we have many more plots. It's a lot more to work with here, right? And you can see that each one of these plots represents the distances from some cage to the cage listed at the top of the title. Distance from samples in C31 to C31, distances from samples in C35 to C31, C42 to C31, so on and so forth. And as we look at this y-axis, remember, higher numbers here indicate dissimilarity. They indicate difference, distance. And lower numbers indicate similarity. So these we would expect to see less distance between the samples in cage 31 and the samples in cage 31. But there is clearly a pattern here where some of these plots, I'm going to zoom out a little bit so you can see them all together. Some of these plots show up with much higher levels of unweighted unifrac diversity than others. In fact, the trend seems to be that they split right down the middle. There's a group of 3 in each of these visualizations that is different from the other group of 3. That doesn't mean a whole lot statistically yet. I'm kind of just looking at pictures and trying to figure out what I see. But fortunately, we also have the statistics to answer those questions in a quantitative way. Because we passed the p-pairwise argument, I'm sure you remember, to the beta group significance visualizer, we have a whole new table at the bottom of our visualizations. And that table lists the pairwise comparisons between each group and each of the other 5 groups, right? So here we have a comparison between cage 31 and cages 35, 42, 43, 44, 49, etc., each with its own sample size, permutations, pseudo-F, p-value, and also new here, a q-value. Just like in alpha group significance, this is a Benjamini-Hochberg correction. You'll notice that these q-values tend to be higher than the p-values because they have corrected for multiple testing bias. We see the same thing over in our weighted unifrac cage significance. Similar visual trends, a little less distance perhaps here. We still have an uncorrected significant p-value for all of the comparisons. But it's probably more interesting to start asking questions about what these pairwise results might mean. And if we step back over to our tutorial, we'll see that we actually have a really well put together question here. From the metadata, we know that all of the mice with transplants from a given donor were co-housed with other mice transplanted from that donor, right? Which means that all of the C31, C35, and C42 mice have the same donor, and all of the C43, 44, and 49 mice have the same donor. So we might expect a significant difference between cages where the donor is different. And this visualization wouldn't account for that. So we can go back in and look and see whether donor is potentially a part of why we see such a strong difference between these samples, or these groups rather. I have some little paper notes here so I can remember what our numbers look like. C31, C35, and C42 were all from one donor. And if I look to the right, I see that our Q value comparing C31 and C35 is not significant. We don't see a lot of difference in weighted unifrac between those 2 cages that have the same donor. The same lack of significance is true between cages 31 and 42. As soon, though, as we get to differences between cage 31 and 43, 44, or 49, we start to see a higher level of difference, all 3 of these being statistically significant. And so maybe what we're seeing here is not so much difference based on cage effect as it is difference based on the donor who contributed to the mice in each of those cages. As such, I would say that the results do look the way that we would expect, especially having seen such a strong level of significance when we looked at the donor influence the association between donor and unifrac distance. So there is one other box we kind of have to check here when we're working with permanova. And the tutorial does a really good job of explaining that. Essentially, permanova differences can be caused either by large differences between groups or by differences and variances within groups. And so in order to know that our statistically significant differences are meaningful, we also need to confirm that that those differences are not just an artifact of within group variance. We can use beta group significance again, but using a different method this time, permdisp instead of permanova, to really quickly check whether we are dealing with that kind of confounding variable in our test. I have, once again, copied the command, pasted it into our terminal, hit enter, and hopefully we'll have a new visualization shortly. Maybe I'll hit enter again. All right, we're in business. We'll refresh. I don't actually remember what this one's called. This is why typing in your own commands is useful. So we are looking at unweighted unifrac cage significance disp this time. Do the old copy paste, and we can once again see all of our box plots. What we're looking for here, though, really is summarized well in our summary table at the top. What we want to know is whether there is a significant difference in variance for any of the cages. And this p-value here at 0.19 is high enough to tell us that there is no statistically significant difference between cages, which is great. It means that we've ruled out a high dispersal, I think it is, dispersion rather, from being the cause of our significant permanova results. This brings us to the last command in our beta diversity tutorial section. This is Adonis, and I am once again going to copy paste, and while this runs, we can talk about what's being passed into this command. Here we are once again taking in a distance matrix. This is just a CHIME2 artifact of the type distance matrix. This one was again produced by Core Metrics Phylogenetic hours and days ago. The same metadata file. It will produce this visualization that we've called unweighted Adonis. And just like we saw back in the alpha diversity section, we're using an R-style formula here. And again, just like in the alpha diversity section, I would encourage you, if you don't understand these R-like formulas, to look at the documentation. So if we head to docs.chime2.org and check out our available plugins, go down to diversity, and we are looking for Adonis. We have a nice description of how model formulas work. Now, Adonis is another multivariate method, and here we're using our formula to try to answer the question, once we take interactions between donor and genotype into account, are either or both of those factors still statistically significant contributors to beta diversity? I think by now our visualization has finished running, so I will refresh this and we'll take a look at what we've got. If I can find it. There we go. Copy our link. Open up Q2 View. And this, refreshingly, is the simplest visualization probably of the entire day. It is a lovely statistical table that hides a lot of really beautiful and complex mathematics underneath it. And all we need to worry about here are the numbers. We'll use those numbers to answer the last question for this section, which is really exciting. It's been a long session again, and we've covered a lot of ground. One of the things that keeps coming up, right, is this idea that donor has a very strong signal in our data. And like we saw in the conversation about cage effect earlier, it's possible for a factor with a strong signal to kind of outweigh other factors in how we perceive diversity in a system. So using a multivariate tool like Adonis lets us kind of tease apart those differences. Here, we want to know if we adjust for donor in using Adonis, do we still have an effect from genotype? And if we do, what percentage of variation does that genotype effect explain? I am not a statistician. I will never be, try as I might. But this is a pretty straightforward table. We have listings for the degrees of freedom, the sum of squares, the mean squares, all of which are used in calculating this F statistic, our F model, right? We have R squared, which we'll talk about in just a moment, and we have the probability that this model could generate a more extreme F. This is, again, a p-value that allows us to kind of quantify how likely it is that we got this result. I'm sorry to all of the statisticians in the audience while I butcher your discipline. So if what we want to know is, after adjusting for donor, whether genotype has an effect, all we need to do is look for the genotype factor in row 1 and note that we have a solid significant p-value here. So genotype retains a statistically significant effect even once we tease out any influence from donor. And it explains, and this is where R squared comes in, R squared describes the percentage of the overall diversity that is explained by this factor. And genotype explains 4.7% roughly of our unweighted unifrac. That doesn't sound like a whole lot, but stepping back for a moment, think about the complexity of a microbial system, right? You have so many different potential influences. So unless you're working with like a prepared environment, there are probably many, many, many different factors that go into or that contribute to how this community is structured, right? We here are trying to look at things like genotype and donor. We've controlled for species, and we have these animals in a controlled environment, but there's still a lot of different factors that come into play here, right? And so it's very common to see low R squared values. And though 4.7% isn't a tremendous amount of difference to explain, we'll take it. This is going to be just fine. This is a publishable result, probably, assuming everything else about our analysis goes well and our experiment was well designed. So we have answered our last question here, and really this is one of the most fundamental questions we're asking here. If this study was intended to test whether genotype significantly influences, or at least is significantly associated with, changes in the microbial community composition or characteristics, we have a really strong result here showing that, yes, indeed, even after factoring out donor, which is a kind of another major covariate, we still have a statistically significant difference coming from genotype, and a relatively solid percentage of that difference explained by genotype. So as I said, this has been a really long session. I hope you all are holding on okay, but we've done some really cool things here. Among them, we got to play around with emperor plots in depth, really see what some of the powerful things you can do with those visualizations are, and start using them to really understand trends in our data that we might not have seen without a good visualization. We got to use a few new commands, CHIME diversity beta group significance and CHIME diversity Adonis among them. And beta group significance we got to use in a few different ways, looking at pairwise differences, using a dispersion test to essentially check our own work. And in the end, we found out that there are significant differences by both donor and genotype in our community's beta diversity. Thank you all again for spending some time with me here. I hope this has been helpful, and feel free to ask any questions that you might have using the question system we've got in place. Have a great day and enjoy the rest of the workshop.
Welcome back, everybody. Thanks so much, Chris, for that fantastic walkthrough there, wrapping up our beta diversity discussion. We have a handful of fantastic questions that came in the general channel while that video was playing. So we're going to take a couple of minutes and start to talk through those. I am joined here in the control room by Chris and several other instructors as well. And so we're going to cycle through some of these questions here together. So I'm going to send this first question your way, Chris. We have a question here that says, if I have a QZA and QZV files for Unifrac and I'd like to look at the PCOA plot, but exclude one category of samples, how can I do that?
So that's a really cool question because there are kind of two answers, or maybe even more. Imagine yourselves looking at an emperor plot with a whole bunch of kind of groups of samples plotted. And you have, for example, a set of control samples, 2 or 3 samples that were specifically put in to check for sequencing bias or something like that. You just want to take those out of your final publication. It is possible to use the, I think it's visibility tab in the emperor plot. And there's a good conversation about this in general, to drop out those samples. You have to be a little careful when you do that, though. If you're not removing kind of a trivial set of samples, because removing samples that have been used in calculating where all of the points in your plot are placed can essentially mean that your emperor plot is not telling the whole truth about how differences are represented. There might be points that are influencing the spread, or samples that are influencing the spread of your plot that are just no longer visible. And so you're kind of lying with statistics there. And the best practices, if you want to take points or take a group of points out of a sample, are to filter those points out using one of the filtering tools visible in the filtering tutorial. And once you have filtered the points out, then you can calculate your distances, your PCOA results, and build your emperor plot so that it accounts for only the groups that are present in the plot itself.
Awesome. Yeah, thanks so much, Chris. Instructors can drop a link to the filtering tutorial into general. That would be really helpful. So specifically, we would be filtering our feature table prior to running the beta diversity metrics, or in this case, the core metrics phylogenetic. Let's see. I have a question here for Justine. So in that discussion there, we looked at unweighted unifrac and weighted unifrac. And we have a question about like what do we do if we see some kind of separation in one of the unifracs, but not in the other?
Sure. So this is one of my favorite things. Keep in mind that yesterday, Ariel and Yoshiki talked about how unweighted unifrac considers the presence and absence of organisms, whereas weighted unifrac accounts for their abundance. And so we're looking at two different aspects of the data, and specifically, unweighted unifrac with presence and absence gives us a better sense of rare organisms, whereas weighted unifrac gives us a better sense of more common organisms. There's also some really interesting stuff about where it focuses on the phylogenetic tree. I'll pull that article and try to put it in the general chat too. But essentially, if you're seeing a difference in unweighted unifrac, but not weighted unifrac, it might tell you that it's a shallow phylogeny thing, or it might tell you that it's a rare taxa thing. Whereas if you see a difference in weighted but not unweighted, it might be deep in the tree or it might be abundance. One of the tricks you can use to do that is to actually add a third metric like Bray-Curtis. And Bray-Curtis should tell you, is it the weight? Because if it's weight, Bray-Curtis should also show you separation. But if it's the phylogenetic tree that's driving the difference, then actually Bray-Curtis would work there too, because Bray-Curtis doesn't include phylogeny. So basically bring in multiple aspects of your data and think about the things that the metrics are telling you, how they're the same and how they're different.
That's really great advice. Thank you. Okay, so the next question that I have here is for Bod. And let's see, the question here is, can you run an interaction term in this analysis? Yeah.
Yes. So when we're talking about the tools that are available in QIIME 2, the Permanova, which is the default test during the beta group significance action, that doesn't have it doesn't have any sort of interaction terms that you can include in the formula. It is designed to take in one grouping variable. The advantages of it is that it has a default pairwise comparison, so a post hoc pairwise comparison. Your best bet, if you wanted to include these terms, interaction terms, whether it's random or fixed, you can use the Adonis plugin that I believe Chris covered in the video. The Adonis is a wrapper for the R package vegan Adonis, and it's Adonis 1, not the most recent one that was released. And that does allow for a formula that it would include interaction terms. Now, one thing I did want to mention with regards to Adonis, because it's definitely important and very often overlooked, is that the order in which you include your terms in the formula is important. So basically what it's doing behind the scenes is that the formula will take the first variable that you put, and it tries to explain the data or the variance using that first variable. And whatever remains with regards to residuals, whatever remains is used by the second variable to try to explain it. So when you see these different values in sequential order, they are explaining of what is remained in your formula. So if you have a scenario where your first variable explains everything or a vast majority of the variance, then even though the other variables might be important, they might not show as significant because the vast majority of the variance was already explained by an earlier variable. So that's something I've noticed a few times people overlook that they're not aware of. I'm not sure what Adonis 2 does, but that's just specifically for the one in QIIME right now.
Thanks for that, Bod. Let's see. My next question in the queue here is for Ariel. So there was a question. So for these unifrac analyses, the Emperor visualization doesn't do the analysis by metadata group, but then our beta group significance visualization does. Do you have a second to comment on that for us?
Hi, I would love to comment on that. So one thing you'll notice if you check the output directory from running core metrics, you get 3 different files for each of those beta diversity metrics, right? So you get, for example, an unweighted unifrac. You have an unweighted unifrac distance matrix.qza, an unweighted unifrac PCOA results.qza, and an unweighted unifrac emperor.qzv. And remember, the difference between QZA and QZV, right, is A is artifacts. They're like tables usually. And QZV is a visualization. That's what the V stands for. So what you're talking about there and what we used for emperor, right, is the unweighted unifrac emperor.qzv. And kind of like we talked a little bit about yesterday, that is specifically a visualizer. It's not a statistical test. So, right, it's like what's behind me here. You can see grouping and clustering patterns between the 2 groups, but you don't actually know if they're statistically significant or not unless you run a statistical test associated with them. So you might use that QZV to kind of start picking up on patterns or to visualize these patterns like in a publication or presentation, but you don't actually know if they're significant yet. So then we can instead use that distance matrix.qza to calculate those statistical differences. So if you remember from my presentation yesterday, that distance matrix is that really, really big table that compares the distance between every single one of your samples with every single other one of your samples. That's like usually too big to look at yourself. So that has all of those differences in it, and that is what you use for the statistical test. So if you, I know it's kind of hard because we're usually just copying and pasting, right? But if you actually look at the command for the beta group significance, what you're using isn't that PCOA results or the emperor plot as your input distance matrix, and you can see there just from the name of that parameter that it's going to be a matrix. So your input is the weighted or unweighted unifrac distance matrix.qza. So that's actually what you're using, and that doesn't really necessarily relate to the emperor at all, if that helps.
Excellent. Thank you, Ariel. Chris, you're up next. I've got 2 questions for you back to back here. The first one, how did you zoom out from your figures when you were giving your presentation?
That is a great question with a really simple answer, fortunately. There's no magic here. You opened QIIME2 View in the Chrome browser, and there are a few ways to tell your browser to zoom out so that you can see all of those different group significance plots at the same time, which can make it a lot easier to interpret. I used the control button and the scroll wheel on my mouse. You might use command and the scroll wheel on a Mac, I'm not sure. And if you don't have a scroll wheel on your mouse, you can use control and the minus key on your keyboard, or you can go up to the 3 dots icon at the top right-hand corner of your Chrome browser window, click on that, and then go down to the zoom controls, which will let you zoom in or zoom out.
Great, thank you. Yeah, that's a really, really useful little tool to have in your tool chest, especially when giving presentations.
Ooh, I've got one more point there.
Oh, yeah.
When you zoom out, I believe this is true in Chrome as well. I know it is in Firefox. In the address bar, you'll see a little representation of how zoomed in or zoomed out you are. So if you're 60%, it means that you're showing 60% like zoomed out. And if you click on that button, it will bring you back to the normal like zoom level. So you don't have to think about zooming in and zooming out much. You can just zoom to where you want and then click on that percentage and it'll put you back at normal.
That's great. Thank you. Control room, I'm going to skip a question here and go down and ask Chris about the, there was a question about output of the beta group significance was unweighted unifrac, but the input was weighted unifrac. Chris, can you comment on that quickly?
Dark black magic. No, I'm going to guess that if the input was one thing and the output was another thing, that's just an artifact of the making of the video. If you put unweighted unifrac into one of these visualizers, you will get unweighted unifrac out. And if you put weighted unifrac in, you will get weighted unifrac out. If you ever run into a situation where that doesn't happen, please let us know.
Well, thanks. Justine, I have a question for you. Is it advisable to use the Q value instead of the P value?
It depends. So the Q value is multiple hypothesis corrected. So we've accounted for the fact that when you do a random test, if like I take a bunch of, if I take dice and I'm asking, what's the probability I see a 6? It's 1 in 6. If my p-value is 0.05 and I have data, then by random, 1 out of 20 times, I should see a significant signal, whether or not the signal's there. And so we penalize that with the Q value, which then lets us say, okay, we've run, 100 tests at random. If it's 1 in 20, then 5 tests should be significant. Personally, I tend to sort of vary on the Q value. Because permutation uses it's a permutation test. So you're already looking at changes in your data. I tend to be relatively confident in a p-value that is 1 over the number of permutations, which is the minimum p-value you can get for a permutation test. I'm a lot more wary if I run 1,000 permutations and my p-value is 0.01. So there were 10 permutations that were more extreme than my distribution of my data. That said, the other reason that Q values are hard is because they may require you to run more permutations, which can get expensive. I think standard in the field is I don't think there is a true standard. I think you will have an easier time with statisticians, with people sort of outside of the microbiome field if you do in fact correct. And so I guess having answered all of that roundabout, try to use the Q values.
Thank
Can
you.
I add one quick thing there?
Of course.
I often just report both. So I'll say this was my p-value for the test. This was my corrected p-value, which is the Q value. And so I don't know, I don't think it's a standard by any means, but I'll often report a statistical test as test statistic, p-value, Q value.
Can I jump back on with that? Because I really like that.
Yeah.
The other thing that makes me crazy when you're doing permutation tests is list the number of permutations you ran. So P 0.001 with 999 permutations has a very, very different interpretation than P equals 0.001 for 9,999 permutations. And so just like when you're publishing, when you're reporting, when you're thinking about it, please, please, please report your permutation number.
Yep, I couldn't agree more. Just want to throw that in. Very important.
Evan, you want to chime in here real quick?
Yeah, just one quick thing. So an example of where you definitely could ignore the Q value entirely is if you have a very specific pairwise hypothesis. And even if some other pairwise comparison has a really great p-value, if you just ignore it and do not consider it, then you can totally just use the p-value. But you need to be kind of rigorous about that.
Thank you, Evan. Bob, did you there?
No, I think I align very well with what Evan was saying, is that for me, the Q values, especially the default Q values for something like beta group significance, is because it has to take into account every single pairwise comparison, it can be quite punishing. So as what I do when I do things custom in R, I have a priority pairwise comparisons, as Evan says, and so I don't penalize myself so much with all of that redundant, unuseful comparison. So it sort of falls somewhere in between. But yeah, I think everything was said was perfect.
Great, thank you. And Bod, while I have you here, we have time for one last question before I think we need to move on. And we still have several great questions related to this topic. We're going to go ahead and put them in our queue for this afternoon's general question and answer session. But Bod, I have a question for you here. Should we ideally be doing all of these permanovas or permdisp and Adonis tests on both unweighted and weighted unifrac, or what should we be doing here?
I don't want to sound like a broken record here, but it really depends on the biological question you're asking, which I think has been the theme of a lot of our answers today. Is, they're designed with a specific purpose. They give you different information, and so to just pick all of them or pick the ones that are significant, but actually have a prior sort of question in mind and then just answer those. Yeah, it really depends.
Great, thank you. Yeah, that is definitely the theme of the workshop. It's QIIME 2 online. It depends. So on that note, thank you so much to all the instructors joining in here, and thank you everyone in our Slack general channel for the fantastic questions. We still have several more that we didn't get a chance to look at just yet, but we will circle back on these at the end of the day. So with that, I want to announce and set up here our next video segment. So Ben Kaler is going to give us a chat about taxonomic assignment, taxonomic classification, and we will probably need to split this video over the break. So I think we're going to break in roughly 10 or 15 minutes here, but we'll get the video started. So with that, turn things over to Ben. Thank you.
Hi, I'm Ben Kaler. I'm going to talk to you about taxonomic classification. I will cover what taxonomic classification is, how to do it in QIIME 2, and a few ways that you might make your taxonomic classification more accurate. So, what is taxonomic classification? We will assume that at this point in the pipeline, you have obtained a set of denoised sequences. So these sequences are probably the output of data2 or dblur, and so you might be calling them ASVs or SOTUs. I'll just call them sequences. So you can just use those raw sequences if you're interested in getting quantitative about the similarities and differences between samples. But if you want to actually figure out which critters are present in your sample, then at some point you're going to have to figure out where these genetic sequences came from. And that's what taxonomic classification is. It looks at each one of these sequences and it tries to figure out which of a range of species, depending on your application, that sequence came from. So the range of species that you might be comparing your sequences against depends on your reference database. So several good reference databases exist and more are being created all the time. Two classics are Silver and Green Genes, and they look like these reference databases here. So it looks a lot like your sequences. It's just a FASTA file inside one of the two database files, which is a feature data sequence QIIME artifact. And it contains a reference sequence and a label for that reference sequence. And then there's another file called, which is a feature data taxonomy artifact. And for each of the sequence labels from your sequence file, it will hold taxonomic information. So in this example here, you've got 4 reference sequences, and each one has a string that categorizes it in terms of its taxonomy. So usually they start with the kingdom or domain, and then the phylum, and then so on down through usually 7 levels, including genus and species at the bottom levels. So in a perfect world, we could just take the sequences from our feature data sequence artifact that we've obtained from Dada2 or dblur, and we could search for that sequence inside our set of reference sequences. And then when we find a match, we could go, okay, well that matches this sequence with this label. Then we could take that label and look it up in the taxonomy file, and that would tell us which species that particular sequence came from. Sadly, in the real world, it's more complicated than that because we have natural variation and we have noisy sequencing. So it's fairly rare that you'll get an exact match between the sequences and the sequences in your reference database, not to mention the fact that maybe the particular critter that was in your sample might not be in your reference database. So we tried to come up with the nearest match. And this is what taxonomic classification is all about. So we get, say for instance, our sequence from our set from our experiment. It's called feature 5 in this example, and our taxonomic classifier might compare it with a range of sequences in our reference database, and it might say, well, actually it doesn't match any of them spectacularly well, but it matches most of the bacterial sequences really well, so we'll call it a bacteria. And for feature 5 here, it matches all of the proteobacteria quite well and better than the other phyla, so we'll call it a proteobacteria, but more than that I'm not really confident to say. And depending on how good a match the taxonomic classifier gets, it will classify each of the sequences from your experiment down to a particular level. So for instance, for feature 2 there, it's done it's more confident and it's gone down 4 levels, 4 ranks in your taxonomy, whereas for features 1, 4 and 5 it's only managed 2, and for feature 3 it's only managed 3. Okay, so how might we go about matching the sequences from our experiment with the sequences in our reference database? Well, we need an algorithm. Now, because you're watching this video, you're probably a biologist or a bioinformatician, so you've probably heard of BLAST. And you're probably reasonably familiar with this concept of sequence alignment. So one way that you can do that, which might make intuitive sense to you, is that you can take each of your query sequences, the sequences from your experiment, and you can align them or blast them against your reference database. That's certainly one way to do taxonomic classification. I'll talk about that a little bit more in a moment. But more commonly, we use what's known as a machine learning classifier. So a machine learning classifier in this case will take your sequence and it will output a classification. And by classification, I mean a taxonomy for that particular sequence. So at the user level, we don't really care too much about what the algorithm is inside of that machine learning classifier, so long as it can take our sequence and give us a taxonomy string out the other end. But I'll tell you about some of the common features of how this problem is approached. So a machine learning classifier takes a set of features and it outputs a classification. So for a sequence of DNA, You might say, how do we get the features out of that we give to our machine learning classifier? Well, we start by writing out our sequence up the top there, as we have in bold, and then we take the first 7 nucleotides, GAC, GAAG, and that's called a 7-mer. And then we move across one position and we take a second 7-mer, and then we take a third 7-mer and a fourth 7-mer. They're called 7-mers because there's 7 nucleotides. In general, they're called K-mers because there might be K nucleotides. So the way that we extract features from our sequence is we look at which specific K-mers are present in the sequence that we're interested in. And the presence and absence of those K-mers are the features that you feed to your machine learning classifier. As a user, you don't have to worry about that too much. This all takes place inside the QIIME2 feature classifier, taxonomic classifier. So you might think of it as we take a bag of K-mers, we feed it into our machine learning classifier, and it spits out a classification like it has done here. So I think there are 6 levels there. So in the way that we usually set it up, that means that the machine learning classifier has taken this bag of K-mers and it's managed, or with some level of confidence, it can say, okay, it's actually the Geobacter genus of this particular taxonomy, but it wasn't confident enough to call its species. So the next question is, well, where does the machine learning classifier come from? Well, when we create a machine learning classifier, we say that we train it. And we train a machine learning classifier by showing it examples where we know the answer. And of course, the examples where we know the answer are our reference database. So we take our long list of sequences from the reference database, and we take all of the taxonomic strings that are associated with those sequences, and we tell the machine learning classifier, if you see a sequence like this one, you should be outputting a taxonomic string like this one. And turns out there are certain algorithms that can learn reasonably well to go from a bag of K-mers to a taxonomic string. The most commonly used one is called naive Bayes. Don't be tricked by the title. It's not really from the Bayesian school of thought in statistics. It's not really from the frequentist school of thought in statistics. It's a machine learning algorithm. Okay. So once we've got our taxonomic classifications, we can proceed on downstream in our pipeline, and we can come up with plots like the one here on the right. This is a bar plot that shows the relative frequencies across a bunch of different samples that are grouped by sample type. And in this particular case, we've asked it to display the frequencies down to the phylum level. But because it's a QIIME2 visualization, not in this presentation, but if you go on to look at these in the tutorial, you'll see that this is actually an interactive plot, and you can select the taxonomic level down to which you want the plot to be rendered. It can sort it by different criteria, you can filter, you can do a bunch of different stuff where you can select particular taxa and it will show you what it's doing specifically across the plot, which is really kind of nice. And once you've got it looking how you want it to look, it will output your plot in a publication quality file that you can then go and include in your publication.
All right, welcome back. Sorry for the kind of abrupt termination there. We're going to break for our first break of the day, and then we will resume and finish Ben's discussion on taxonomic assignment. So let's see. It's yeah, we'll be back in 30 minutes exactly. We'll try and give you a few minutes heads up. Thank you. Welcome back, everybody. We are going to resume Ben Kaler's lecture on taxonomic assignment, and I think the control room is probably going to rewind that video maybe about 30 seconds. And then at the end of that, we'll have some time for a few questions. And with that, I'll turn things back over to Ben. Thank you.
It's a machine learning algorithm. Okay. So once we've got our taxonomic classifications, we can proceed on downstream in our pipeline, and we can come up with plots like the one here on the right. This is a bar plot that shows the relative frequencies across a bunch of different samples that are grouped by sample type. And in this particular case, we've asked it to display the frequencies down to the phylum level. But because it's a QIIME2 visualization, not in this presentation, but if you go on to look at these in the tutorial, you'll see that this is actually an interactive plot, and you can select the taxonomic level down to which you want the plot to be rendered. It can sort it by different criteria, you can filter, you can do a bunch of different stuff where you can select particular taxa and it will show you what it's doing specifically across the plot, which is really kind of nice. And once you've got it looking how you want it to look, it will output your plot in a publication quality file that you can then go and include in your publication. So, like I said, the most common method is to use a machine learning classifier, but you can actually use BLAST or a similar program, or a program that has similar functions called vSearch, and that will let you compare the sequences that you got from your experiment with the reference sequences, and it does a thing called consensus classification. So for each query sequence, it will look at the BLAST hits, for instance, that it gets in the reference database, and it will look at that list and it will say, in this example here, we've got a sequence, all of the hits were a bacteria, so we'll call it a bacteria. In fact, all of the hits were a proteobacteria, so we'll call it a proteobacteria. And all the way down until we say, okay, all of the hits were Legionellacea, so we'll call it that genus, but not all of them were Legionella. So we won't say what species it is. Now you can adjust the sensitivity and various thresholds for that. You can, when I say consensus, you can actually make it vote amongst the hits that it gets, or a certain number of the top hits, to see which one is the majority, and then so you can adjust the confidence of it in that way. These algorithms work almost as well as the naive Bayes classifier, and if you're not happy with the naive Bayes classifier, or you think it's playing up in some way, then you can try these consensus classifiers, or even if you're just worried about the risk that using a particular piece of software is biasing your results in some way, then you can try out these consensus BLAST and consensus vSearch methods to give you a second opinion on your taxonomic classification. Finally, people have been doing this for a long time, longer than QIIME2 has been around. So if you do have a taxonomy from the past, then you've got it, for instance, in a biome file, you're able to import that into one of these feature data taxonomy artifacts, and then you can use it downstream in the same way that you would have done had it been generated by the QIIME2 feature classifier, taxonomic classifier. Right, that's an overview of what taxonomic classification is. Now let's start to get into the nitty-gritty of how this fits in with the QIIME2 ecosystem. So this graph here, this flowchart, is fairly intimidating. There's a lot going on. I'm not expecting you to take it all in the brief time that I will show it to you. You can go and look at it on the internet if you want to really pore over it. But these two bits here are the interesting bits in the sense that they are related to what we've been talking about so far. So down the bottom left here, we've got the query sequences. They're the sequences that you've gotten from your experiment. The little rounded rectangle that says taxonomic classifier is the taxonomic classifier that we've already trained, and you can see from the way those flows are coming together that we combine those in the feature classifier plugin we call classify sklearn with that taxonomic classifier and our query sequences as inputs, and it will output a set of sequence classification results. From there, you can use those to filter your sequences or go downstream and create all the nice plots that we've seen. You could even do differential abundance analysis if you wanted to. Alternatively, you could use the consensus classifiers there. They don't need a pre-trained taxonomic classifier. They work straight off the raw reference databases, which you can see being imported on the top left. Now, we actually offer pre-trained taxonomic classifiers for your convenience on the QIIME2 website. So if you go to the QIIME2, I'll show you the link in a minute, but if you go to the QIIME2 website and then go to the documentation, you'll find some data resources that contain some commonly used pre-trained taxonomic classifiers. So if you're using 16S or you're using V4 in the V4 locus in the 16S gene, and you aren't interested in using anything other than green genes or silver, then I'd recommend that you go online and you can find that taxonomic classifier and you can go ahead and do your taxonomic classification. There is a caveat to that, and that is that with those classifiers, using those standard techniques, you can probably be fairly confident that you're going to get reasonable classification down to the genus level. If you want to go all the way to the species level, then you probably have to work a bit harder. And we're going to talk about that in a second. So if you want to do that, you're going to have to train a taxonomic classifier for your particular problem. Alternatively, if you aren't using 16S, if you're using 18S or ITS or CZI, then you will have to train a classifier on a reference database that you have obtained externally to this process. So if you want to do that, you have to go to this part of the flowchart, and you've got your reference taxonomy, your reference sequences. You'll import those into feature data taxonomy and feature data sequence artifacts, and then you will feed them into feature classifier fit classifier to train a taxonomic classifier. There's an additional step in there that's labeled trim reference sequences. Now, I think in most of the cases where you're doing this yourself, I'd probably advise against skipping that step. So you can take the feature data sequence data from your reference sequences and feed that straight into the feature classifier training step without going through the trim reference sequences. We found that trimming reference sequences to a particular amplicon region of interest was mildly beneficial for, say, 16S v4, but if you've got a novel database that you're using and you're not sure, for instance, that all of your primers are present, or there are other complicating factors, then I'd happily advise you to skip that trim reference sequences step. Just take your reference taxonomy, your reference sequences, and then go ahead and train your taxonomic classifier. Okay, so let's go a little bit deeper into what's happening in this orange rectangle here and why we might want to do that. Oh, sorry, before we do that, this is just a link out to the data resources. So there's some of those pre-trained classifiers. There's a little bit of a warning that comes along with that. If you are using a pre-trained classifier with our feature classifier plugin, then you should make sure that it's coming from a trusted source and you're downloading it in a secure fashion using, say, HTTPS, and you should probably check your checksums. I know that's not really what checksums are for, but it can't hurt. Now, the reason for that is that if someone really wanted to hack your system and somehow they figured out that you were using these and somehow could impersonate somebody you know, then it is possible to inject malicious code into those pre-trained classifiers. But you know, I'm calling that a fairly remote possibility. Just make sure you get them, you either train them yourself or you get them from someone you trust in a way that you can trust. Now, if you want to learn more about training feature classifiers, then we'll talk about that a little bit in the tutorial, I think, or there are online tutorials, and there's a link to here to a tutorial in the QIIME 2 documentation. You can find that pretty easily just by going to the QIIME 2 documentation and putting in taxonomic classification. It'll be one of the top hits. So, why might we want to use something other than the standard reference databases? Well, there are two issues with making reference data. Well, actually, there's many issues that come with making reference databases, but one of them is that it's a big job. And while you might hope that every classification in your reference database looks like this ideal 16S here, where you know where it is in your taxonomic tree right down to the strain level, in reality there's a bunch of sequences in most reference databases that look like this, where they haven't been classified for whatever reason, even past genus, and the species is a not particularly informative number labelled by OTU. Right, so these things exist right through most reference databases, and it is ongoing work of a large number of dedicated people to produce better reference databases. And if you want the latest and greatest, then maybe you might want to try one of the novel reference databases that are becoming available. A second issue is that, well, I said it's a big job, it's also a hard job. So, for example, look at this sequence here. This is the first 90 nucleotides of the V4 region in 16S for Lactobacillus helveticus. It is also the first 90 nucleotides of that same region for Lactobacillus hamsteri, right? So two different species, exactly the same sequence. This is kind of a toy example that I have inserted here to illustrate a point, but it is actually a symptom of a much larger problem where you can have two quite different bacterial species, so different even at the genus level or above there, that are almost identical genetically. So how are you going to get around the fact that the data that you're using to pick between species might not be able to tell you the difference between those two species? Well, one way that we can do that is to bring in additional information. So, for instance, I downloaded data from a 2002 study where they took 3,921 human vaginal samples, and I performed taxonomic classification, and I found that Lactobacillus helveticus accounted for 21% of the reads across those almost 4,000 samples. Now that's huge. That shows you that there's really quite low diversity in these samples. The fact that one single species accounts for 21% of all the genetic material that we saw. On the other hand, Lactobacillus hamsteri was a bacterial species that was isolated from a hamster gut in 1987. not known to be associated with human vaginal samples. So, if you saw that exact sequence in a human vaginal sample, would you call it Lactobacillus helveticus, or would you call it Lactobacillus hamsteri? Well, you'd be able to make a pretty safe guess that it's Lactobacillus helveticus. The point that I'm trying to get to is that if you know where a sample comes from, that this problem of a single read possibly belonging to multiple different species is made much simpler. It turns out that there are many fewer overlaps of a single genetic sequence or very close genetic sequences in terms of their taxonomies if you're sampling from a specific habitat. So you can use that information. One of the ways that you can use that information is that for several different habitats, you can download a custom database, right? So there are custom databases for human vaginal samples as well, and for several other habitats that people are specifically interested, you can download databases for those specific habitats. And they have been shown to be able to increase the accuracy of your taxonomic classification past the genus level down to the species level and possibly beyond. We've taken a slightly different approach in QIIME 2, and I'll show you this fairly complicated slide here. The approach that we have taken is to say, okay, when we usually train a taxonomic classifier, so supervised learning is the particular type of machine learning that we're interested in here. When you train a taxonomic classifier, the standard approaches implicit in the assumptions that are behind those standard approaches are that you're actually equally likely to observe any of the classifications that you might output. So if you look at green genes, for instance, it might say that not green genes. If you use the standard taxonomic classifier that we make available under the data resources, for instance, there is an assumption in the mathematics behind that that says, well, actually, if we have to guess between hamsteri and helveticus, it's a 50-50 split. We can do better than that by looking at the distributions of species across samples for a particular habitat type. So that's what we did, and we created a tool. So I and the floating head of Nick Bokulich there wrote this tool called Clawback, which plugs into QIIME and can query the Cheetah database, which is an online database of many thousands of microbiome samples, and it can build up a profile of what the taxonomic distribution is for a particular type of habitat. So for instance, if you're interested in stool, you might look at animal distal gut. For instance, we used the Earth Microbiome Project ontology habitat classifications. So you can pull out a particular set of what we call taxonomic weights. So for animal distal gut, for instance, you've got a particular distribution where it says, okay, you're more likely to observe some species than some other species if you're looking at animal distal gut. And then you can feed that as an input when you're training your taxonomic classifier. So that all sounds a little bit complicated. We've tried to take some of the hard work out of that by making a repository of taxonomic weights that we've trained on cheetah samples and from some other sources at this repository called Ready to Wear, which is in my GitHub account if you want to go and have a look at that. So you can download those. You can train them against usually green genes or silver, and then you can go on to use that taxonomic classifier to get better accuracy in your taxonomic classifications. Does it work? Yes, it works. So we did a bunch of rigorous testing where we trained weights and classifiers on a particular set of samples for a particular reference database, and then we tested those classifiers on samples that hadn't been used to generate the weights or the classifier, and we found that if you are using taxonomic weights from a habitat and then you go to classify samples drawn from a habitat like that, then it's always going to improve the accuracy of your taxonomic classification. So for instance, here's a data set that Nick Bokulich had, where previously with a certain level of confidence, the classifications, the taxonomic classifications were down to the genus level there. So you can see that's on the top plot here, where you've got Enterobacteriaceae and Bifidobacterium and Bacteroides. And on the bottom level, with the same level of confidence, using these taxonomic weights to train our taxonomic classifier, we were able to say, okay, actually we can say it's E. coli, or B. adolescentis, or B. ovatus. And if you're interested in your actual taxonomic classification, you're probably interested in which specific species that you've got in your sample, whether in fact it came from a hamster or from a human. Okay, that's it. So if you would like to know more, please look at the tutorials online. There's tutorials for how to do taxonomic classification using the standard classifiers, how to train taxonomic classifiers, how to use clawback. And if you have any questions, please don't hesitate to ask more on the QIIME2 forum.
Welcome back. Thanks so much for that lecture there, Ben. I am joined here in the control room by Ben Kaler and also the one, the only, Dr. Nick Bokulich. We've got one or two questions coming in here. We have time to address those. So I'm going to send this first question to both of you here on the call. So after our taxonomic results are obtained, I'm interested in some specific features, ASVs or OTUs. Is there some way that I can track back in order to figure out what features or sequences were lost in previous steps?
So I could take the lead on this, Ben. So the short answer is no, not necessarily, not in an easy, straightforward way. If I understand the question correctly, you have your taxonomy results and you want to find sequences belonging to that taxonomy that were lost in the process through filtering or what have you. But once they're lost, you don't know what they are. That's kind of the point of taxonomy classification, that you've carried the sequencing data along through to this step, and after you perform taxonomy classification, then you know the annotation for those sequences. But if they've been lost earlier on, they don't survive to that step. So the best thing I think you could do in this case is you could take a representative sequence for that taxon, and then prior to filtering your data, prior to quality filtering or wherever you're losing sequences, you could try and use some kind of similarity search tool like vSearch to try and find sequences that are similar to that representative sequence within a certain similarity threshold.
Thanks,
Nick. That's a great answer. That actually helped me to understand the question. But the only thing I would add to that is that standard bioinformatics advice, which is if you want to know how your decisions at earlier steps are affecting your results, try varying those decisions, so changing the parameters that affect those filtering steps to see how that affects how many of each taxi you're getting through at the final stage.
Thanks for that, Ben. Ben, I've got another question here for you. Is clawback convenient for use when exploring a new ecosystem or environment?
So you do need a certain number of existing samples in order to figure out some reasonable set of weights for that environment. I think we went as low as about 150 when we were doing testing, so 150 samples. But all is not lost. You can still use what we term the average weights. I can't remember whether I went into this in the presentation, but you can, we've made available on a resource called Ready to Wear. I'll drop a link for that in the general chat. But you can access weights that are averaged across a wide range of different sample types that were found in Cheetah. And generally that gives better classification results than using the standard uniform classifier so yeah, if you're using a novel environments, I'd say use the average weights if there's nothing else that compares to it. Otherwise, getting something that's fairly close can also help as well. We showed that the closer the environment is, the environment type is to the environment that you're actually sampling from, the better your results. But just trying to get something that's close can work as well. So I guess like everything, it depends, but there's a few options that are available to you there.
Great, thanks, Ben. I've got one more question that just came in here, and I'll send that at you or at Nick, whichever one wants to jump in here. What do you think about 16S rRNA gene copy normalization?
Nick, I'll let you do that. You've probably got a more nuanced understanding than I do.
So I think it's a good thing in theory if you have some kind of sophisticated tool that's able to do it correctly. But the literature I've seen on this topic, at least, has shown that the tools that are out there often don't perform especially well, just because unless if you have a really well characterized environment, then you might not have representative genomes for all of the organisms that are detected. And so you might not be able to correct for a gene copy number in any given environment. Additionally, within different species, there tends to be a certain degree of variation in there. So again, the literature I've seen on this topic has kind of shown poor performance. This would actually be a good topic for general discussion on the forum, by the way, because I think we've discussed this on the forum before, and I think there have been some interesting discussions on this topic. Anyway, my kind of feeling on it is also that additionally, across bacteria, you don't have that much difference in 16S copy numbers. It's something like an order of magnitude at greatest. But by and large, I believe most bacteria are somewhere between 2 to 6 copies in general. The kind of second and third quartiles are somewhere around there. So all in all, what I'm trying to say is that it's not going to be a life changer to correct for copy number normalization. It of course is going to give you more accurate results, but it's not going to be a total game changer. Additionally. This is something that's going to affect your data across all samples. So it's going to be a way to try and better reflect the relative abundances of different species if that's something that's important to you. But in something like differential abundance testing, it's probably not going to matter.
Bod, did you have something you wanted to add to that?
Yeah, just something I read about recently, which I thought was really nice and relevant to hear. Everything Nick said, I 100% agree. One of the issues with these normalization tools for the copy number normalization As it turns out, it was because they suffered from a lot of metagenomic bias. So by not accounting for the metagenomic bias that's been recently sort of highlighted in the literature, these tools didn't really perform well. But as soon as those metagenomic bias correction tools were applied, a lot of these normalization for copy number tools appear to be doing really well, actually. So as Nick said, it's not something right now that's super available or reliable, but I think there's been breakthroughs in that regards, and soon, hopefully within the next year or two, they'll be quite readily, widely used and available.
Great, thank you for that. So we are going to take a quick pause on the Q&A here and resume the discussion on the taxonomic classification. Anthony Simard is a undergraduate in the Capareso lab, and he's going to give us a presentation on performing taxonomic assignment and looking at like taxonomy bar plots and so on. So we're going to shift over to that. Keep your questions coming in on general. We will have time for more taxonomic assignment question and discussion at the end of that section. So with that, take it away, Anthony.
Hello, my name is Anthony Samar, and in this video we will be discussing the taxonomic classification portion of the CHIME 2 Parkinson's mouse tutorial. This video will generally assume you've been following the tutorial videos up to this point, and also that you've watched Ben Kaler's lecture video on taxonomic assignment, because in this tutorial video we will be discussing how to implement some of those concepts in CHIME 2. So to start with, you can see I have these 3 files in my current working directory. These 3 files are what you will need at bare minimum from previous portions of the tutorial to complete this portion of the tutorial. If you've been following the tutorial up to this point, you will have these 3 files and more, and you will be fine. If you're trying to hop right in on this portion of the tutorial, you will need to go download these from previous portions. And these are our feature table sequence, our feature table frequency, and our tabular metadata, respectively. So, moving on to the tutorial itself, we have taxonomic classification. For this analysis, we will use a pre-trained naive Bayes machine learning classifier, which you may remember from Ben Kaler's lecture is perhaps the most common way to identify different taxa. This particular classifier was trained to differentiate taxa present in the 99% green genes 13.8 reference set trimmed to 250 base pairs of the V4 hypervariable region. Now, why do we care about that? We care because naive Bayes classifiers perform best when they're trained for the specific hypervariable region amplified. So if you're using a naive Bayes classifier that was trained on V4 data, or rather if your data is V4 data, you're going to want to use a machine learning classifier that was trained on V4 data. If you have V4 data and you use a classifier that wasn't trained on V4 data, you're not likely to get good results. So just keep that in mind when you're doing this. Make sure the classifier you use matches the data that you have. So let's actually get that classifier by using this wget command in the workshop server terminal. And you can see that went off fine, and we now have our classifier. And the next thing we'll do is run the command that's really the meat of this portion of the tutorial, and that's CHIME feature classifier sklearn. This will take our feature data sequence, the classifier we just downloaded, and it will produce a feature data taxonomy artifact. Now that artifact will contain all of our ASVs with different taxa assigned to them by the classifier. So I will run this command, and I strongly suspect this command will take probably more time than all the other commands in this portion of the tutorial combined. It really is the meat of this portion of the tutorial, and in the real world, using a real full-size data set, this one could take some significant amount of time. I mean, you can see here, we're using a fairly small sort of toy data set for this tutorial so that things don't take too long, and even then, it's taking a minute. So you can see we now have our feature data taxonomy saved to taxonomy.qza. But you might remember from previous portions of the tutorial or some previous experience that these QZA files, these CHIME artifacts, aren't really meant to be human readable. They contain the data in a format that is easily understood by computers and not so much by people. So in order for us to interpret that data, we're going to want to turn it into a visualization. Hence the next command in the tutorial, CHIIME metadata tabulate. This takes our feature data taxonomy artifact and it outputs this visualization that will allow us to actually view the data in a more human readable form. And this should run significantly faster than the last command, and you can see it did. So in order to actually view this visualization, I'm going to go over here to this web page that is currently looking at the current working directory on the workshop server that I'm running this tutorial in. I'm going to refresh the page so that it gets back all of the contents that have entered that directory since the last time I was here and loaded this page. And I'm going to copy this link address, go to CHIIME view, go to file from the web, paste that URL, and go. And you can see we now have our visualization. This visualization contains feature IDs, the taxa associated with those feature IDs, and the confidence with which our classifier was able to associate this taxa with this feature ID. You can sort this any way you like: ascending, descending feature ID, ascending, descending taxon, ascending, descending confidence. I'm going to keep it on ascending feature ID, which is the default. You can also see here we have a search bar. This search bar can be used to narrow down the data in here, as you might imagine. So let's say I just want this particular feature ID, plop it in the search bar, and now that's all we have. Let's say I want only things that are in the phylum Firmicutes. Type it in the search bar, press enter. All that's left are taxons or taxa that are from the phylum Firmicutes. You can do the same with confidence, but it doesn't work maybe in quite the way you'd expect. If I were to put in, say, 0.9, well, we basically have everything with a confidence of 0.9 and up because these all contain 0.9. But if I were to put in 0.90, now we only have things that contain 0.90 explicitly in any one of these three columns. In this case, given that I put in 0.90, it's only really going to be in confidence. So it's not anything 0.90 and above, it's things that have 0.90 in them. So don't think you can put in, say, 0.98 to get everything with a confidence of 0.98 and higher. It won't do that. It'll give you everything with a confidence between 0.98 and 0.99. So, moving on to the next portion of the tutorial, we're going to want to visualize some more of our data. In particular, we're going to want to visualize our feature table sequence. And so we're going to do that using feature table tabulate seeks. We're going to run that, and it's going to be exactly the same as before. We're going to take in that artifact, we're going to output a visualization, we're going to go to our page that has the workshop server directory open in it. We're going to reload. You can see that we now have this data2repset.qzv. No, I do not want this page in Catalan. It is not in Catalan, rather. We're going to copy this link address, CHIIME view, go here, file from the web, paste, go, and we have our visualization. This one contains the feature IDs of all of our features, the sequence length, which, as you can see, is 150 for all of them, and the sequence itself with a hyperlink on it. And we'll see what this hyperlink is for later. So, going back to the tutorial, we have some questions to answer. First, find this feature. What is the taxonomic classification of this sequence? What's the confidence for the assignment? So, if you learned from before, you might guess that what we want to do is copy this feature, go back to our taxonomy.qzv, and search this feature. And you can see that we have this particular taxon associated with this feature ID. And we'll discuss what this means in a moment. The fact that this is just a G double underscore and an S double underscore as opposed to an actual genus and an actual species. You can also see that the classifier was about 98.36 so on percent confident that this feature ID belongs to this particular taxon. This number here might be a little bit different for you, not a huge deal. So that's how you do that. Let's clear this search bar and go back to the next question. How many features are classified as G. acromantia? So what we're going to do is double click this, copy, paste it in here, and you can see we have 2 G. acromantia features. And in fact, these are both classified as the species Mucinophila with a very high confidence. So you can see we have these 2 features, both the same taxon, both very high confidence. And at this point, I think it is worthwhile to address these notes because we've now seen both of them. First, you might notice that some features do not have taxonomic assignments, which for the Green Genes database is indicated by a blank string at the level, e.g. G _. These indicate that there is not enough information for the Green Genes database to differentiate members of that clade, either due to ambiguity in the database or because the gene region being sequenced doesn't provide the resolution to distinguish members of that clade. So if we go back to our visualization and we get rid of this, you can see, here we have family, genus, and species, _. Let's actually resort by feature ID low high. I don't know why it wasn't sorted correctly there. My apologies. But let's look at this example instead. So we can see we have genus and species is just G __ and S _. So what that means is that the database itself doesn't have enough data to determine what genus or what species this particular feature was. It could get it to kingdom, phylum, and class, order, and family. Genus and species, it doesn't know. And there is another kind of missing data, which is something like this. You can see we have a genus, but we don't even have an S __ for species. We just have nothing. That comes from the case where Q2 feature classifier cannot reliably classify the ASV to a deeper level. In those cases, incomplete taxonomy strings will be provided. So, in other words, the ones where we see the level __ come from the Green Genes database not having enough data to differentiate that taxon to that level. And the ones where we don't see the letter or __ at all means that Q2 feature classifier with your given classifier wasn't able to assert what that was at that level. The next note, you might notice that one ASV has the same taxonomic assignment as another. That is perfectly normal, and in fact we did see that with G. acromantia here. Unique ASVs do not necessarily map to unique taxonomic groups, which makes perfect sense. If you're gathering a bunch of data, especially if you're gathering a bunch of data from the same kind of area, you might get data from the same thing, and that's perfectly fine. So, now that we've addressed those 2 notes, let's move on to the third question. Use the tabulated representative sequences to look up these features, that being the G. acromantia features. If you blast them against NCBI, which I will show you how to do you get the same taxonomic identifiers you obtained with Q2 feature classifier? So, I'm going to get this back. I'm going to go back to our visualization. I'm going to search it again, and you can see we still have these 2 feature IDs with this particular taxon. So, in order to blast it against NCBI, what I'm going to do is I'm going to take this feature ID, I'm going to copy it, I'm going to go over to our rep set QZV, and I'm going to use the browser search, which is Control F. I'm going to paste that feature ID in, and we're going to click on this hyperlink on the sequence, which will bring up this page. And as you can see right now, NCBI blast is having some server issues. So I actually ran these commands beforehand, so I will be able to show you the output, but I'm not going to run them live here. I'm just going to show you how you would run them. So again, that's find the ID in this QZV that came from your feature table sequence. Click on the hyperlink on the sequence, and it'll take you to this page, and you would click View Report. Now again, we're not actually going to do that because the servers are down, and also it can take a significant amount of time to run these commands. I actually ran them earlier before the servers went down, and it took close to half an hour. So depending on the load on the NCBI servers and whether the servers are down for maintenance, etc., you know, all of that can affect the degree to which you can do this. But just to reiterate that those steps, we'll look at the second feature ID from that same taxon. So again, we will copy the feature ID, go over to our other visualization, paste it in the search bar. Here we are, here's the sequence, click on the sequence, takes us to the NCBI BLAST page. This is what it should look like if the server is working properly. This is the server being bad. This is what it should look like if it worked. And again, you would click View Report and wait for results. And now I will show you what those results look like. So as I said previously, I ran it before, and this is the output from that first one, from that first feature ID, clicking on the hyperlink on the sequence, and then clicking on that View Report. This is what it came up with. So if we scroll down here, we can see what different taxa BLAST associated with. And also you might remember BLAST again from Bing Kaler's lecture. Basically what it does is it's taking your sequence and comparing it to a whole bunch of sequences that are already sort of known. And whatever it has the most in common with, whatever known sequences has the most in common with, that's what BLAST is going to tell you it probably is, which makes sense. If we look down here, we can see that some of these don't really have a name that we care about, but if we move down a bit, we can see Acromantia municiphala. And in fact, if we go over to the results for the second feature, we can again see Acromantia municiphala. So yes, NCBI BLAST did give us the exact same taxon for these features as QIIME2 feature classifier did. So, moving back to the tutorial, the next portion is the taxonomy bar chart portion. Since we saw a difference in diversity in this data set, we might want to look at the taxonomic composition of these samples. To visualize this, we will build a taxonomic bar chart of the samples we analyzed in the diversity data set. Before we do this, we will first filter out any samples with fewer features than our rarefaction threshold, which was discussed in a previous tutorial. In this case, our rarefaction threshold was 2000. So we can filter these samples out by using the Q2 feature table plugin with the filter samples method. That is going to take our feature table frequency, our minimum frequency, in this case 2000, and is going to output a new artifact of feature table frequency that has been filtered. So we're going to run that to get this table2k.qza, which again is pretty much the same as data2table.qza, only any features with less than 2000 sequences have been removed. Or rather, any samples with less than 2000 features have been removed. My mistake. And again, we're going to want to create a visualization out of this artifact so that we can actually view the data. This visualization is going to take the table we just generated, our new feature table sequence. It's going to take our feature data taxonomy that we generated before using classify sklearn, and it's going to take our metadata. So let's run this. I'm going to clear because this is getting a bit messy. Paste and enter. And we will have a new visualization. And there's actually only one question associated with this one, so we'll look at the question a bit before we go look at the viz to motivate why I look at the viz in the way I do. So the question: visualize the data at level 2, phylum level, and sort the samples by donor, then by genotype. Can you observe a consistent difference in phylum between the donors? Does this surprise you? Why or why not? So let's go look at that viz and see if we can answer this question. So I'm going to go back here. I'm going to reload the page again. Taxabarplot.qzv is the viz we want now. I'm going to copy the link address. I'm going to go to QIIME view. I'm going to click on this. I'm going to get a file from the web, paste, go, and we now have this visualization. And the first thing I'm going to do is to change the taxonomic level from level 1 kingdom to level 2 phylum, because everything we have is in the kingdom bacteria, and maybe a tiny bit of unassigned here, so this isn't very interesting to look at. But if I change it to phylum, now we have more variety. The next thing I'm going to do is take this bar width slider and slide it over as far as I can without moving any samples off the right. And you can see that blew up the viz and made it much more easy to look at. So let's go look at that question again. Sort by donor, then by genotype. Can you observe a consistent difference in phylum between the donors? How do we do that? Well, sort samples by over here. Click on it. We have donor. We have genotype. So if I click on donor, we can see that everything is still pretty dominated by Bacteroidetes, also has significant numbers of Firmicutes. But if we look at this HC1 donor here, this one is the only one that really contains this Actinobacteria phylum. And if I go to genotype, we can see it's really only the susceptible mice here that contain this Verrucomicrobia. The others are fairly well spread out on genotype, but Verrucomicrobia is only in one wild-type mouse and is fairly prevalent in some of these susceptible mice. So this can give you a good idea of where to start with your analysis. It's not, the end-all be-all of what to do. This doesn't necessarily mean anything significant. Your gut feeling, my gut feeling, anyone's gut feeling looking at this viz does not replace the need to do some real statistical analysis, but this might be a good place to start that analysis. And also worth mentioning, we can do some more fiddling with this visualization. So let's say you want to publish this in your paper, but you don't like the way it looks. We can change the color palette around. Maybe you prefer one of these. That one's a bit interesting, a bit hard to read. I kind of like the default one. We can also sort ascending or descending, and so on. So that answers this question, I believe. And remember that the answer to this question does not necessarily give a definitive conclusion for your study. I hope you learned something here today. I hope this was of some use to you, and I hope you will use QIIME 2 in the future to do some taxonomic classification for real-world projects. Thank you.
Welcome back. We've got some excellent questions that came in from you all. And I have a big group of instructors in the control room with me here. So we are going to start working through some of these. So let's see. Let me pull up my list here. Can you remind me again about this the question from Marco here in the control room? I'm sorry, I lost track of if we were. Okay, we're going to jump on to the next one. So Marco had a question here that I think got put into our queue and then has been addressed in Slack. So I've got a question here for Greg. And the question is, should I be using a rarefied table when I'm generating my taxa bar plot?
Yep. So quick answer to that is no, don't use the rarefied table when you're generating your taxa bar plot. You should use the unrarefied table, but usually what you might want to think about doing is filtering samples with low read counts from that table before generating it. The reason you don't want to do rarefaction first is because part of that process is generation of relative abundances from the feature counts. And so the samples are normalized that way. But if you end up with, a few samples in there that have very low read counts, like maybe 10 or 100 or 200 reads, those would probably end up getting dropped from your diversity analyses by rarefaction, but you would not be able to differentiate them necessarily in the taxa bar plot. You can do that with the QIIME feature table filter samples action. And I'll post a link to that, and I'll post where you can find an example of that in the tutorial in response to your question.
Thanks for that, Greg. The next question that I have here is for Ben. Ben, what about some of the other taxonomic classifiers, such as random forest, XGBoost, and others? Could you talk us through your thoughts on that?
So yeah, that's a great question. Naive Bayes is an older technique, and it's really simple. And the surprising thing is that it frequently, well, maybe not frequently, but it sometimes really punches above its weight in terms of the performance that you can get out of it. And it has an advantage that it's simple to understand, and it's relatively fast to train and can work with quite large data sets. So we actually wondered that ourselves, though, whether maybe random forest or some other methods, more advanced methods, can improve taxonomic classification. And we've done some testing. We haven't published it yet, but the early results are that it's actually really quite hard to beat naive Bayes in this space. And certainly we weren't able to get performance out of those other techniques that was consistently better than naive Bayes. So we're sticking with it for the moment.
Great, thanks for that, Ben. Control room, I'm gonna scroll down to the last question in here, and I'm going to send that to Mike. Mike Robeson, are you around?
Yep,
so
I am here.
Great, thanks. So this question here is someone noted that we use the 99% green genes database, so 99% OTU clustered, on our ASV table, which is, we've kind of mentioned before, we've sort of thought about this as analogous to like 100% OTU clustering. What's going on here? Yeah.
Sure. So there's a couple reasons, more reasons than what I'll briefly outline here. But largely, it's an issue of practicality. So a lot of these reference sequences, right, they don't necessarily come from high throughput sequencing. There's no form of denoising. They're either usually from older Sanger sequencing reads. They might have some issues. And there's a lot of redundant information in a full database. So we do clustering of some sort. A lot of the databases will actually do this for you. For example, as you've noted, Green Genes provides a 99% cluster. Silva also provides a 99% cluster. I responded to a very similar post like this on the forum. If you want to search the forum for why not use database identity, you'll see a post which I responded to that describes this, and also the reasons why even Silva, for example, uses their NR99 reference database. Also, this reduces the database size. Many of you have probably run into issues of, say, using a Silva classifier or any large database classifier like cytochrome oxidase, and you could run out of memory, or you need a high memory machine. So another benefit of clustering your reference data is to reduce that database size so that you can effectively search it. If you want to make your own 100% identity database, we provide some tools and ability for you to do that, which is outlined in the post. Hopefully that answers your question.
That's great. Thanks so much. Does any of the instructors have anything else they'd like to add in here before we shift gears and start thinking about differential abundance? I'm seeing some head shaking. Okay. Well, thank you so much, everyone, for the great questions, and thank you to our instructors here for taking some time to address those questions. So with that, we're going to move on into our next topic here. This is differential abundance testing. And our presenter on that is Jamie Morton. And Jamie is at the Center for Computational Biology at the Flatiron Institute, Simmons Foundation. And with that, we are video queued up. Thank you very much.
Hello, I'm James Morton, and I'm a researcher at the Simmons Foundation. Today I'm going to talk about a concept known as differential abundance, a technique that's commonly used to identify microbes of interest. In the previous lectures, we've talked about how to design experiments and how to perform analyses such as alpha and beta diversity. But oftentimes, when we're trying to interpret our data sets, we want to be able to see more than just clustering patterns of our biological samples. We also want to be able to identify microbes that could be explaining the patterns that we observe in alpha and beta diversity.
Imagine for a moment that we're interested in utilizing 16S sequencing data to investigate obesity in mice. We don't need to build a new diagnostic for obesity. We can tell which mouse is obese just by looking at it. However, determining if there is a microbial difference between these mice is far more interesting, since that could provide hints at some of the mechanisms underlying obesity. Identifying which microbes have differing abundances between these mice can give us even more hints. This is the goal of differential abundance. In practice, performing differential abundance can be tricky. Let's consider another example. Suppose we're on a boat in the ocean and we're interested in studying ocean microbes. We collect a bucket of seawater and count 6 orange and 6 blue microbes. But shortly after this expedition, there was an oil spill. We rush out on another expedition to collect another observation to now count 8 orange microbes and 4 blue microbes. The question is, given these observations, what can we infer about what happened to these microbes? Did the blue microbes decrease in abundance? Or did the orange microbes increase in abundance? One possibility is that the blue microbes halved after the oil spill. Another possibility is that both microbes increase in abundance. Both of these scenarios will give us the same proportion of microbes. The only difference between these two scenarios is the difference between the microbial totals, or the total microbial biomass. Our collected buckets cannot distinguish between these two different scenarios since we don't know how many microbes there are in the entire ocean. If we want to answer the question of identifying which microbes have changed in abundance across our experimental conditions, we need to have some knowledge of how much the total microbial biomass has changed. If we're lucky, we can estimate this quantity by estimating the volume in our system and use instruments such as qPCR, spike-ins, or flow cytometry to estimate the microbial cells per unit volume. However, if we cannot obtain a good estimate for the volume of the system, inferring the total microbial biomass and differentially abundant microbes becomes more complicated. If we cannot infer the total microbial biomass, then what can we infer given our measurements? To better understand this, let's try to reformulate this model in terms of simple mathematical terms. Let's encode the microbial abundances in environment A and B, as shown here. These are the true abundances in the environment for the microbial species. To determine which species have changed, all we need to do is compute the ratio of each microbe between the two environments. If this ratio is equal to 1, that implies the microbe abundance is not different between the two environments. If this ratio is greater than 1, that implies the microbial abundance is greater in environment A compared to environment B. And vice versa. If the ratio is less than 1, that implies the microbial abundance is less in environment A than in environment B. But remember, we often cannot directly observe the microbial abundances in the environment. Instead, we need to sample from the environment. From these samples, we can estimate the proportions of the microbes in this environment, and we can separate the proportions that we can measure and the total microbial biomass that we cannot observe, as shown here. With this in mind, if we try to calculate the microbial differences between the two samples, we will end up with this resulting expression. The change in the microbial abundances depends on both the change of proportions and the change of total microbial biomass. In other words, the inability to measure the total biomass will bias our estimates of the log-fold change.
So there you have it. Unless you're able to measure cell concentration and the total volume of the system, you cannot determine for certain if a microbe abundance has changed. Now the question is, what can we infer given the data at hand?
In order to figure out what we can infer, we need to be able to ignore the change in total microbial biomass. One way is to take the classic chemistry approach. Similar to how concentrations are computed by taking water as a reference, we can try to identify a reference microbe. The ratio of two microbial abundances will act like a concentration, and the total biomass dependence disappears. Since these ratios don't depend on the scale of the total biomass, these quantities are said to be scale invariant. If we revisit our previous example, I may not be able to tell you if the blue microbes or the orange microbes have changed in abundance, but I can certainly tell you that the ratio of the blue to orange microbes has changed from 1:1 to 2:1. Both of the different scenarios that we were struggling with before express the same change in ratios. If I choose the orange microbe to be in the denominator, I have chosen this microbe to be my reference frame, and this concentration will measure how much the blue microbial abundance has changed relative to how much the orange microbial abundance have changed.
So now the question is, how do we identify a meaningful reference microbe? There are many ways to do this, which we'll cover throughout the course of this lecture. To help us with the process of identifying these references, we will discuss another scale invariant concept known as ranking.
The idea is that the microbial biomass bias applies to every species equally. While we cannot determine which microbe has changed on the absolute scale, we can sort the microbes according to how much they have changed. We refer to this procedure as differential ranking. Consider this example. Here we have two microbial populations before and after an event. If I compute the ratio between these environments for each of these microbes, I won't be able to tell you which microbe has increased or decreased in abundance. However, I can tell you which microbe has increased the most or decreased the most. If you're running statistics to identify which microbes to probe and culture, these are the statistics you want to focus on. Ranking these microbes by how much they have changed can help you and your colleagues prioritize which microbes to focus on.
We have provided a high-level overview behind the idea of differential abundance analysis and highlighted some of the conceptual challenges when identifying differentially abundant microbes. Furthermore, we've shown how ratios and ranks can help alleviate some of these issues when answering biologically relevant questions. Next, we'll provide some intuition on how to perform differential abundance analysis using generalized linear models. Previously, we talked about the concept of differential abundance and how ranks and ratios can be useful. Here, we're going to talk about generalized linear models and how almost every differential abundance method is built on top of this concept. But before we do that, we need to do a quick recap on analysis of variance and how linear regression is connected to this concept.
The main idea behind analysis of variance, or ANOVA, is we want to determine if there is a difference between two or more groups by testing to determine if their means are different. For now, let's consider a simpler scenario. Suppose that we are only interested in observing differences in body mass index between two different populations. Here, we have collected measurements from two populations, a diseased population highlighted in orange and a healthy control population highlighted in blue. We wish to quantify the difference between these two populations with respect to their body mass index. To do so, we will denote the body mass index as a variable y, and we'll label the sick samples with x equal to 1 and the healthy samples with x equals 0. Here, x is a variable that determines if a patient belongs to a healthy group or a sick group. If we take a peek into this data set, we can see a body mass index measurement and a group label for each row in the table. Here, each row is a unique measurement for a patient, and we'll add another column which we'll call x, and we'll replace the sick labels with 1s and the healthy labels with 0s, and we'll denote the body mass index with a variable which we'll call y. If we want to quantify the difference between the body mass index measurements, between these two groups, we can use linear regression of the form y equals to beta naught plus beta 1 times x. Since this is linear regression, we can tune the intercept beta naught and the slope beta 1 to get the best model fit. Once we have identified the best values for beta naught and beta 1, it can be shown that beta naught is the average body mass index for the healthy patient, and beta naught plus beta 1 is the average body mass index for the sick patients. You can see this for yourself. If you set x equal to 0, you will retrieve the average body mass index for the healthy patients, and when x equal to 1, you will retrieve the average body mass index for the sick patients. This also implies that the difference between these two averages is given by beta 1, the quantity we're actually interested in. The process of encoding these groups as zeros and ones are typically referred to as contrasts, because we're interested in comparing or contrasting these two groups. The cool thing about this approach is that we can easily extend this to multiple categories. Suppose I want to compare the body mass index across 3 different groups: a healthy group, a disease group without treatment, and a disease group with treatment. To encode this as a linear regression, we can create 2 contrasts. For instance, I can look at the difference between the healthy group and the disease group without treatment, in addition to the difference between the healthy group and the disease group with treatment. To encode these contrasts in a form that the computer can understand, we can represent these variables as zeros and ones. For the first contrast, C1, we can place a 0 for patients that aren't in the disease group without treatment, and a 1 if they are in that group. Similarly, for the second contrast, C2, we can place a 0 for patients that aren't in the disease group with the treatment, and a 1 if they are. Let's draw up what this regression will look like. Remember, from the previous example, we need to have an intercept, which we'll denote with another column of 1s. Altogether, I will get 3 terms: beta naught, beta 1, and beta 2. Beta naught will give me the average body mass index of the healthy group. Beta 1 will give me the average difference between the healthy group and the disease group without treatment. And beta 2 will give me the average difference between the healthy group and the disease group with treatment. Since we're already dealing with multiple categories, there is nothing stopping us from extending this to multiple covariates. For instance, we could look at body mass differences while accounting for differences in gender. The encoding is set up in the same manner. We can build contrasts for both gender and the disease group. It might seem like a lot of work, and once upon a time, all of these concepts were crafted by hand. But now, we have computers that can automate much of this process. We can use formulas to specify our experimental design to automatically generate our contrast matrix. As you can see, this contrast matrix was generated in one line of code using a software library called Patsy. These formulas can be highly expressive. You can encode continuous quantities, interaction terms, to enable more complex experimental designs. I highly recommend checking out this library for more details.
Now that we have done a quick recap on the analysis of variance and linear regression, we'll switch gears a little bit to tie in these concepts into our data sets. Now remember, our data is made up of sequencing counts. Because of this, we need to explicitly model these sequencing counts using counting distributions. The simplest counting distribution out there is the multinomial distribution.
The probability distribution of the multinomial is given as follows. Let's try to provide some intuition exactly what this is modeling. Let's imagine that we have a huge environment with billions of microbes. If I remove one microbe from the system itself isn't really impacted. If I collect a few dozen microbes, that is still a small fraction of the system. If the sample is sufficiently small relative to the environment that we are trying to measure, then we can make the assumption that the microbial count that we have measured can be characterized by a multinomial distribution. What do I mean by a multinomial distribution? Suppose we are looking at a bucket with just 3 colors. Each color represents a species, and each ball represents a microbial individual. if the proportions of microbes in this bucket are 1/4, 1/4, and 1/2, and if I draw 20 balls from this bucket, I should expect to get roughly 5 red, 5 green, and 10 blue balls. If I repeat this sampling process many times, and over time you may notice it's not very common to get only red balls, only green balls, or only blue balls. Here is an illustration of how these sample proportions can be represented. Each of these corners represents when a sample only observed red, or green, or blue balls. In the center of this triangle, we have a sample given by 1/3 red, 1/3 green, and 1/3 blue. Most of these samples can be found in the center, as shown by this thick cloud of points. The takeaway here is that there's uncertainty around the microbe proportions that we observe. Every sample we collect is not guaranteed to give you the exact proportions of microbes in the environment, just due to the sampling process itself. If we want to make informed decisions about these microbes based off of sequencing data, we need to account for this uncertainty in sampling as well.
So now the next question is, how can we account for this uncertainty? How can we reconcile the multinomial distribution we just learned about with ANOVA and linear regression that we learned about previously? This is where techniques from compositional data analysis can help us.
If we take a look at the distribution of multinomial samples again, you may notice it lives on the strange triangle. This triangle is known as the simplex. It's the space where all proportions live. The proportions in this space don't follow the intuitive real space properties that we're used to. The quantities here are bounded, meaning that every point is composed of proportions that sum to 1. If you try to model these proportions using standard arithmetic, we will quickly run into issues with estimating negative proportions or proportions greater than 1. Have you ever heard of a microbe with negative abundance? Modeling microbes with negative abundances would violate many of the biological laws that we are familiar with.
In order to model proportions using standard techniques such as linear regression, we need a way to reconcile the differences that we just discussed. One way to do so is to have a transformation that can transform proportions into standard real coordinates that we are more familiar with. Fortunately, there are many ways to do this, but I want to focus your attention on just one way, in particular, the center log ratio transform.
The center log ratio transform, also known as the CLR transform, is a function that takes in proportions as input and will convert them to log ratios. The transformation itself can be written as follows. If you recall from the introduction section, this is reminiscent of taking the logarithm of a concentration. The reference here is the geometric mean of the proportions, so you can think of the CLR transform as computing a log concentration, where our denominator here is the average microbial taxa proportion in our sample. Since these log ratios don't have the same constraints proportions do, we can define our linear regression as we normally would. Here, we can predict the microbial log abundances in terms of our experimental design. In this regression, beta naught is proportional to the average microbial log abundances in the healthy population, and beta 1 is proportional to the average microbial log fold change between the sick and healthy populations. And X represents the labels for the sick and healthy samples. Here, both beta naught and beta 1 are d-dimensional, so you can think about this as a linear regression per microbe. If we want to cast our predictions back to proportions, we can also do that. The inverse CLR transform, also known as the softmax transform, can be given as follows. This will later prove to be extremely useful. We can perform our linear regression on log ratios and then use this inverse transform to project this regression back to proportions bounded within the simplex.
It is important to know some of the caveats behind this transformation. The CLR transform cannot handle zeros because the logarithm of 0 is undefined. This is a problem with microbial sequencing data because in the data set, it's mostly dominated by zeros. Next, we'll discuss how linear regression, the multinomial distribution, and the CLR transform can all work together jointly underneath the framework of generalized linear models.
Here is the high-level bird's-eye view of our differential abundance model. We're going to have a model that takes in as input our covariates using the contrast that we have specified. This input will then be fed into a linear regression that is used to estimate the microbial log-fold change we are interested in. The linear regression predictions will then be fed into an inverse CLR transform that will convert these log ratios to proportions. Finally, these proportions will be plugged into a multinomial distribution to predict the microbial counts. The observed microbial counts are then used to evaluate the likelihood, or the probability our observed microbial data could be explained by our model. The model is then optimized. In other words, it is tuned in order to obtain the model that best describes our data. This model will help us answer the biological questions we had while addressing the technical challenges we have outlined. The linear regression component will help us understand the difference between microbes due to the covariates we have measured. The inverse CLR transform will help us model proportions using a standard linear regression. The multinomial distribution will help us model zero counts where traditional compositional data analysis tools will struggle. At this point, we have provided a high-level architecture of how modern differential abundance tools are built. Next, we'll discuss how to choose parameters when running these models, how to evaluate the accuracy of your model, and how to interpret the underlying model parameters. With these models at hand, we need to have a way to evaluate how good the model is. A common metric with linear regression is to use R-squared, which measures the variance in the data explained by your model. Now, there's a real danger of overfitting your linear regression, where you are merely memorizing the microbial abundances instead of actually learning something biologically useful. If your data is overfitted, metrics such as R-squared will not be very informative. We can try to get a handle on overfitting through a technique called cross-validation. The idea behind this is to first fit the model using a portion of the data set, and then predict the microbe abundances for the remainder of the data set. If the error rate of the predicted microbial abundances is comparable to the model error on the training samples, then that is a sign the model can generalize beyond the samples it had observed. One way to measure this cross-validation error is through what's called a Q2 score. This is actually the same concept of R-squared, only applied to the samples the model hasn't seen before. The Q2 score has a similar interpretation to R-squared. A score close to 1 indicates a perfect fit, and a score of 0 or below indicates a very poor fit, indicative that a model has overfitted. If we go back and look at our pipeline, we can rewrite the entire model in just one line, where Y represents our microbe counts that we're predicting, X is our design matrix representing our covariates, and B represents the linear regression coefficients. The question here is, how can we interpret these regression coefficients? Let's go back to the example dealing with two groups, analyzing samples from sick and healthy patients denoted by A and B. If we focus on the regression coefficients for species I and J, it turns out that the difference between these two regression coefficients will give us the average difference of the log ratio I and J between the sick and healthy patients. In addition, these regression coefficients are proportional to the log-fold change. This also means that we can rank the regression coefficients from largest to smallest. So the microbe with the largest coefficient will have the largest log-fold change and will be observed to be increasing the most relative to all of the other microbes. The microbe with the smallest coefficient will be observed to be decreasing the most relative to all the other microbes. We need to emphasize the term relative because we only know the log-fold change up to some constant value. We won't know for certain if these microbes are actually increasing or decreasing. Back to the model evaluation. If you have an overfitted model, what are your next steps? One possibility is to get more data, either from publicly available resources or sequencing new samples. More samples can help against overfitting. Alternatively, you can employ a technique known as regularization that can essentially create a bound on your regression coefficients. Enforcing constraints on your regression can also guard against overfitting. To create a reasonable bound, you need to have some idea about the regression coefficients you expect. For example, if we're looking at differences before and after a dietary intervention, how large of a difference do you expect? Do you expect the microbes to be completely replaced? Or do you expect an order of magnitude difference in their abundances? Suppose that you don't expect more than an order of magnitude difference. Then you can create a very tight bound on your regression coefficients. If we use a normal distribution as a prior and center that around 0, we can shape it so that 66% of the data lies within one standard deviation. This essentially means that for 66% of the microspecies, the log-fold change will be less than 1, if our standard deviation is 1. In other words, we don't expect the majority of microbes to double or half between conditions. Taking this one step further, we expect 99% of the microbes to be within 3 standard deviations. In other words, 99% of the microbes are expected to have a change between 1/8 or 8-fold. If you're more focused on finding a difference instead of exactly estimating the difference, you could be more loose with these bounds. You could even incorporate the resolution of your sequencing to estimate good priors. In the average 16S experiment, there are roughly around 10,000 reads per sample. The logarithm of 10,000 is roughly around 9.2. So this means that for a given pair of samples of 10,000 reads, you can now infer log-fold changes that are greater than 9.2. So if you're not sure what is a reasonable prior to set, setting your prior to a width of 9 or so is reasonable, since it's fairly close to the resolution limit of your machine anyways. Once you are satisfied with your model, then you are ready to start considering hypothesis testing. Unfortunately, how to best perform hypothesis testing is still an outstanding question, but we can definitely perform statistical hypothesis testing on individual log ratios. Here is an example of how such an exploratory analysis will look like in an interactive tool called Chiro. The ranks estimated from the generalized linear model can then be linked to the log ratios where the statistical test can be performed. While much of the content in this lecture is based on the architecture of Songbird, it is important to note the differences across different differential abundance tools. Tools such as ALDEX2, DSeq2, and EdgeR will test to determine if the regression coefficient is equal to 0. In essence, this will test to see if the microbial abundance has changed relative to the average microbial abundance. The underlying assumption here is that the average microbial abundance has not been altered across experimental conditions. ANCOM, on the other hand, will test all pairwise ratios of microbes with the assumption that few microbial species have changed in abundance. Songbird does not perform hypothesis testing, but instead tries to accurately rank the microbes, providing a scaffold for interactive hypothesis testing using tools such as Chiro. There are also tools such as Phylar, Phylofactor, and NICE that were designed to test differences between groups of microbes. This can be especially useful when testing differences between phylogenetic clades. As you can imagine, there are quite a few model choices that can be made within this generalized linear model framework. For instance, the multinomial distribution is amongst the simplest counting distributions that is used in Songbird and ALDEX2, but there are more complex distributions, such as the negative binomial distribution, that's used in DSeq2 and EdgeR, that can better account for technical variability. There are also different choices of prior distributions that can be used. I showed how to use the logistic normal distribution that's used in Songbird. If you use ALDEX2, you'd use the Dirichlet distribution as your prior. In addition, the CLR transform is not the only compositional transform that can be used. There are other transforms, such as the ALR, ILR, and the arcsine transform that are commonly used to transform real-valued variables to the simplex. The choice of transformation will have an impact on the choice of priors, and possibly the interpretation of the regression coefficients. Furthermore, we have not discussed how to estimate model uncertainty in the regression coefficients. There is emerging work on how to perform Bayesian inference that's worthwhile to check out. I have also provided a tutorial on how to build your own Bayesian differential abundance tool.
If you have made it up to this point, congratulations! You've just now obtained a high-level overview of how to perform differential abundance analysis. The good news is that many differential abundance techniques are built inside the framework of generalized linear models, with many of them using counting distributions and some variant of the compositional transforms that we just discussed. If you've understood this material, then you are well equipped to perform differential abundance analysis on your own data sets. I look forward to seeing the exciting discoveries that you will achieve with this newfound knowledge.
Welcome back. Thank you for that lecture, Jamie. We are going to break now for a 30-minute coffee break. And when we resume, Jamie and the other instructors will be standing by to answer any questions. So during the coffee break, if you think of any questions, please submit those into the general channel on Slack, and we will chat about them in about 30 minutes. So we'll try and give you a 5-minute notice on the general channel before we resume. Thank you. Welcome back. I hope everyone had a great break there. So I am about his discussion on differential abundance testing. Jamie, are you around?
Yes, I'm here.
Welcome. Perfect. Like turn on your camera, that's great. So I'll give you this first question in the list here. So one of the attendees would like to know what you think about zero inflated negative binomial models.
Yeah, that's a good question. So for those of you that don't know what zero inflated negative binomial is, it's another way to deal with many zeros in your data. The way it works is that I have a model that's really split into two parts. I have one part that is the negative binomial that allows for the counting of microbes, and then I have the other part that deals with all the zeros. And it's designed for very sparse data sets. The one thing about these types of models is that they assume that there is a mechanism that causes a zero inflation. And in the case of biology, you might think of it that the sequencing randomly malfunctioned and a whole bunch of microbes are just randomly left out. Now, but one of the implicit assumptions of this model is that it means that there could be very high abundant organisms that are randomly masked as zeros. So it really depends on what kind of experimental setup you expect to deal with. So in many of my cases, I don't recommend negative binomial, zero inflated negative binomial, because I don't think that assumption readily applies. But maybe there are some other scenarios where it could be more appropriate.
Great, thanks, Jamie. Yeah, it sounds like one of our common refrains here during this workshop has been the phrase "it depends," and it sounds like this is another case of that. So let's see, I've got another question here for you. What are the available statistical models to study the association of OTUs with the continuous outcomes?
Yeah, so the session that I just discussed was using primarily categorical variables, mainly to make it easier to understand. But you could also imagine that you can extend those easily to continuous valued data. So rather than having, is this sick or healthy, can I look at the pH? So in that case, you can just use a standard linear regression. My tool Songbird can do this, but I think there are a number of other tools that can do this as well.
That's great. Thank you. Any of the other instructors in the call here, do you have any questions for Jamie or any discussion points that you want to bring up? Maybe not just yet. We gotta take some time to think. Oh, maybe Evan's joining us.
Yes, hello. I just wanted to hear Jamie's thoughts on something like a multinomial Dirichlet versus pretty much any CLR style system. So do we transform our data in kind of the Atchison way, or do we rely on basically the Dirichlet being supported on the simplex? What are your thoughts?
That's a really good question, Evan. So so basically the question is, do we want to have a more probabilistic model explicitly handling accounts like the multinomial? Or do we want to just transform the data and then crunch it through our standard linear regression frameworks? The problem with the latter approach is that in order to transform the data, you've got to deal with the zeros somehow. And that can be tricky. So, and I'm gonna just say that it's currently outstanding discussion on how to properly transform data. Me personally, if I'm given the chance, I do try to model it in a more probabilistic setting. But there are scenarios where it's not easy to do this. It's one of the scenarios where it's hard to say if there's a clear winner or not, because it really depends on the situation.
Great, thank you. Let's see. I've got another question here in the queue for you. What do you think is the best way to deal with zero counts in transformation? Pseudo counts or model-based approaches?
Yeah, yeah, so this is kind of goes, it kind of writes off the argument with the zero inflated models. It really depends on what assumptions you're comfortable with walking in with. So when you're dealing with, for instance, they call them rounded zeros in the sense that microbe is in such low abundance that your sequencing instrument might not be able to detect it. So these are the sorts of scenarios that pseudocounts are designed for. But then the question is, well, what type of pseudocount should you be using? There is a much more extensive discussion in the compositional data analysis literature on trying to learn reasonable imputations. I'll say this, kind of going off of Evan's comment about the Dirichlet multinomial, is that the pseudocounts can be troublesome, but if you can use a multinomial-based model, that can sort of deal with these zeros in a much more efficient manner.
Great, thank you. Well, it looks like there aren't any more questions in the queue. So I think with that, we're going to jump into our next video segment. This is presented by Evan. It's a continuation of the Parkinson's mouse disease tutorial. We're going to look at differential abundance. I think specifically we're going to look at ANCOM here. Then we'll be standing by for more questions and discussions afterwards about differential abundance testing. And I believe Jamie will be here with us then still as well. And then we'll end the day just like we've done the other days in this workshop with a general question and answer time. And also, actually, never mind. We'll just move on into that video now. Thank you.
Hello, my name is Evan Bullion. I'm a PhD student under Greg Capareso at NAU. I'm also a research software engineer and one of the lead developers of the QIIME2 project. Today we're going to be talking about ANCOM, and to do that we're going to start with me kind of drawing out a sketch of what's going on with a pretty simple example, and then we're going to return to the tutorial and follow that. So suppose we have two samples. So we have sample 1 and we have sample 2. And within these samples we have features. So we have features A, B, C, and so on, all the way through F. Similarly, in sample 2, we have the same 6 features, all the way to F. And if we're interested in differential abundance, we might be interested in how, say, feature A in sample 1 changes in sample 2. And so let's suppose that feature A actually increases in sample 2. Well, a consequence of this is all of the other features in sample 2 must decrease. And I'll explain why that is in a second. Similarly, if sample 2's feature A were to actually decrease instead, then that would mean that all of the other features in sample 2 must increase. So the key to understanding this behavior is each sample has its own sequencing depth. And so sample 1, I'm going to say the sequencing depth is in 1. Similarly, for sample 2, it is in 2. And so what this means is that all of our features, so let's say feature A plus feature B, and so on, up until feature F, which is our last feature, must equal that sequencing depth. And the same thing is true of sample 2 over here. So if we were to increase A, just like before, that means in order to still equal N2, so this is still N2, we have to decrease all of the other terms in order for that equality to still be the case. And so we tend to not think about this too much because these sequencing depths are something we generally don't control, so they're kind of random. But they do exist, and so our data is constrained by the sequencing depth of our sample. So if we wanted to compare feature A in sample 1 with feature A in sample 2, we have a slight challenge because all of these features are related to their unrelated sequencing depths. So a trick we can use is we can see the frequency of feature A as really being the proportion of A times that depth. So these are equal. And if we look at it this way, we realize that when we take the ratio of something, so let's say we have the proportion of A times its depth over the proportion of, let's say, feature D times its depth. Well, since these are the same term, they just cancel out. And so we can just say that is equal to the ratio of feature A over feature D. And so we can use this to understand change between our samples, which is independent of these sequencing depths. And so over here, we can say something similar. We can say, what is FA over FD? And now we have something we can compare between the two, because we've canceled out that sequencing depth. Now, there's one last thing we might want to address, and that's that the comparison of feature A over feature D is not going to be the same as the comparison of feature D over feature A. And that's maybe not so desirable, but it's a pretty easy thing to fix. So if we take the log of this, so if we say the log of that, well, now the log of the other side is simply its inverse. So we can say that is the negative log of this. And so this is basically our log ratio, and it's what most compositional methods are based on. So if we return to our community, we know we're interested in some kind of ratio. And so we want to know how maybe the ratio between feature A and D changes in sample 1 compared to F versus D in sample 2. And so something we can do is set up a pretty simple ANOVA. And so we can say that our equation looks like the log of FA over FD, and we're going to have some intercept. We're going to have some beta for sample 1, and we'll have some residual as well for 1. And so then we can do the same thing on the other side. So we'll have the log of FA in sample 2 over FD, and we'll have, again, that global intercept, and then the effect of sample 2. So that'll be our beta 2. And then again, some epsilon, which is our error. So now we have something we can test. So we can ask the question, is beta 1, the effect of sample 1, different from beta 2, the effect of sample 2? And so if they're the same, then that means this ratio in sample 1 has not really changed in sample 2. So there's not really a difference here. But if they are different, then that means that we have some kind of significantly different ratio. So we have a differentially abundant ratio. And so that's pretty handy, but it's still a ratio. So we don't know whether or not we can pin the blame on feature A or feature D. We just know the ratio between them has changed. So I've now combined those two communities, and we're going to talk about it in terms of just the features. So if we're interested in, say, feature A to D, if this edge is significant, that means in sample 1, this comparison has a different beta than in sample 2. So let's suppose it is significant. And let's assume that the difference in beta 2 compared to beta 1 is negative. Well, that either implies that F feature D has increased, or it could also be the case that feature A has decreased. We can't really tell those apart. But ANCOM has one more trick, and that is to do all of the comparisons. So, if we were to test feature A against feature B, feature C against feature A against E, A against F, and then likewise over on D, make those same comparisons. So D to B, D to C, D to E, and D to F, we can try and determine which of these is more significant. So let's suppose that over here in feature D, we find the comparison is significant, 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 and significant. However, when we look at feature A, only the connection to feature D was significant. And so who do we think is actually differentially abundant here? Well, odds are pretty good it's going to be feature D. And so if we count up these significant edges, that's going to be our W score. And so here we have a W of 1, 2, 3, 4, 5. So we'd say the W of feature D is 5, whereas the W of feature A is only 1. And if we do that for all comparisons against every single feature, so I draw out this crazy graph, we will have kind of a network and a distribution of W scores. And so with a W of 5, that's basically as good as you're going to get. This is the theoretical maximum for 6 features. But maybe we'll see a W of 4, or maybe a W of 3. And so what we want to do is look at the distribution of these W's, because we're going to expect it will be bimodal. There will be some W's, so some set of features, where the W is awfully high, and then others where it's really not that high, so it'll be low. And that's how we're going to identify which features are actually differentially abundant and in what direction. Okay, so I have the tutorial up here now, and so you should make sure that you scroll to the section Differential Abundance with ANCOM. So one of the first steps we're going to do is we're actually going to filter some features out. So I'm going to copy this command, and I'm going to go over to my console, and I'm going to make sure I'm inside of Workshop Mouse Tutorial, and I am going to paste that in. Oh my goodness. I wasn't expecting it to copy right then. So I'm going to come back over here, I'm going to copy, and I'm going to paste it in. So I'm going to hit enter, and what this is going to do is it's going to take our table 2K. So this is a feature table where all of the samples have at least 2,000 features. And we're going to add an additional refinement. So we're going to drop features which are either seen less than 50 times or in less than 4 samples. And if we wanted to fully understand the definition of that, so whether it's inclusive or exclusive, we can test that by typing QIIME feature table, filter features, help. And so we can see the definitions of these things. All right, so now that I have that, I'm going to return to the tutorial, and we're going to talk about why we might want to do that. So one thing to realize about ANCOM is, since we're testing for differential abundance, it's more useful if the features we're testing that with are actually present in multiple samples. Otherwise, there's really not the opportunity for those to be differentially abundant at all. You'll really just get differential presence absence. And it'll be on features which probably aren't very important because they're either rare members or just noise outright. So that's kind of the goal here. And now, scrolling down a little bit, we're going to do another step. And so this is QIIME composition add pseudocount. And so what a pseudocount is it's a way of kind of getting around the fact that you cannot really divide by 0. And even if you could, taking the log of 0 is also no good. So what we're going to do is we're going to add a small number to every single observation. So where we would have seen a 0 in our table, so for some sample and some feature, there was just a 0 because we didn't observe it. We're going to pretend we did observe it, but only like once. And so that's what this command is going to do. And so essentially what we're doing is we are saying that we have made an observation of almost everything. And so that's going to help us actually take these log ratios. And so we're going to assume that every 0 is not necessarily a structural 0. Rather, it's more of a measurement 0, where our resolution was just too poor. So if we had sequenced deeper, maybe we would have observed something. And so we're just assuming we've observed at least one thing so that we can take a log ratio. It's not the most elegant way to approach this, but it does work. Yeah, I do hope that in the future we come up with some better strategies for this, but 0 is a real problem with compositional methods. Okay, so the first thing we're going to do in ANCOM is we are going to analyze by donor. So we're going to ask the question, so we have these samples, right? We're going to ask the question, are the samples whose donor are healthy versus those with Parkinson's disease, is there a change in any of the log ratios, and is there enough of a systemic bias for one feature, so it's causing many log ratios to be significantly different, that we would believe that that OTU or that feature is the cause of it. Maybe not the cause of it, but the reason that we see a differentially abundant feature at all. So the reason for the differentially abundant ratio. So I'm going to hit enter. And so this is going to take a little bit to run because it is doing all pairwise comparisons between these groups. You'll notice we also ran in that same command a test against genotype. So now we're asking the question, does the predisposition of the mouse to kind of our Parkinson's model cause a difference in any of the OTUs that we observe? And so what we have now are two QZVs. And so what I'm going to do is I'm going to go to workshop server.chime2.org/my username, which happens to be Astute Lobster, because I thought that sounded cool. And you're going to see quite many more files. I only grabbed the files I needed for this tutorial, which is why there's so few. But the things we're interested in are ANCOM donor and ANCOM genotype. So let's maybe take a look at donor first. So I'm going to copy the link address, and I'm going to go to view.chime2.org. I'm going to select file from the web. I'm going to paste that in, and we're going to run. And so the first thing we see is kind of this very interesting plot. So this is called a volcano plot. And when I was talking about the W scores, so the number of comparisons which were deemed significant for the test we ran, I said that we'd see kind of a distribution. And so what we're seeing here is that distribution of W scores. And so what we want to do is find those values which are most extreme. And so there's maybe not a perfect cutoff point here, but it's pretty safe to say that this feature here, which has a very high value of W, is probably more differentially abundant than something down here, which is probably only has a W score of 28 because some other features changed, changing that guy's ratio. Now on the x-axis, we have basically the effect. So you'll see CLR here, and that is the centered log ratio. And so this is a way to aggregate basically the impact of that one feature against the rest of the community. So we're interested in features that not only seem to be awfully suspicious in the number of times they have been found in a differentially abundant ratio, but also those that have an unusually large effect relative to the average. which is all this CLR is telling us. So W is basically the, not exactly the significance, but the importance of that feature. And the x-axis is the effect. So down here we see a table kind of explaining the same thing. And so ANCOM has chosen essentially all of these features as being differentially abundant. And this is the donor group. So these are features that we think have systematically changed between the healthy control donors and the Parkinson's donors. We can also look at percentiles for these two groups. So we have healthy control and Parkinson's donor. Whoops. So you'll notice at the 0th percentile, we have these ones. Remember, that is artificial. We had an imputation step where we added a pseudo count. So every one you see here is actually a 0 deep down. And then we have the 25th percentile. And so now we see for 27, if we look over in the Parkinson's, it looks like this maybe isn't differentially abundant so much as it is differentially present. But that's still interesting. Let's see if we can find a nice differentially abundant one. I suppose this one's okay. So over here, we do have a feature which is pretty rare, it looks like. And it's found more in the Parkinson's group. So this is indeed differential abundance. Just looking through here. Here's a better one. So this one has some actual observations. Again, relatively rare in the healthy control. And also actually pretty rare in the Parkinson's group. But we are detecting, you know, an actual difference in its abundance. So that's kind of what this visualization is showing us. If we look at the other one, so going back over to the workshop server, I'm going to look at genotype because we already looked at donor. So I'm going to, nope, not that. I am going to copy the link address. I'm going to go to view.chime2.org. File from the web. I'm going to paste that in, and that's the ANCOM genotype. And so here we're going to see kind of a pretty extreme change, right? So most of our values, even if they have like a pretty reasonable effect size given the range of our data, really don't seem to be separating themselves in terms of all the log ratios that we are comparing. Something that does kind of stand out are these two. And you'll have to excuse me, that is my cat. She hears me talking and really wants to understand what's going on. So we see this split in distribution that is kind of what we're looking for. And so we see these three features that ANCOM selected, so that would be these top three. And we can see this first one actually is really nice. So we have some real observations of it in the susceptible group. So we're looking at genotype now. So this is our mouse model. And then also actual observations of it in our wild type. But ANCOM has identified this as an actually differentially abundant feature. These last two appear to be more of a presence-absence kind of thing. So we have some observations in the wild type. And again, really nothing going on here. So if we go back to the tutorial, we can think about these questions. So the first question is, are there more differentially abundant features between the donors or the mouse genotype? So if we look at this, we see donor has a great many features that are differentially abundant and genotype less so. And the question prompts us, do we expect this given beta diversity? So if we remember some of the visualizations we looked at before, so I'm just scrolling up here to take a look at beta group significance would be pretty good. I want donor significance. So let's look at that one, and then not seeing a very exciting one for I might cut this out of the video. We'll see. All right, so we have the donor significance, and we see there does appear to be kind of like a difference happening here, right? We have a p-value that looks pretty good. Visually, it seems convincing enough. And so if we look at our donor, I think that's recapitulated given the number of features that are really different here. And so if we have a lot of features that are differentially abundant, we would expect that the distances between these two groups might be a little bit higher with beta diversity. All right, the next question is, here we are. Are there any features that are differentially abundant in both the donors and by genotype? So let's take a look at that. And the way I'm going to do that is I am going to search the small set against the large set. So I'm just going to type that in, and Chrome says there's no results for that. I'm going to do the same thing here. And just to remind people what these crazy strings are, they are a hash of the sequence. So basically we've taken the full sequence of this section of 16S, and we've kind of mushed it up into this little string, which is much simpler to look at. This is called a hash. It's not compression because we have actually lost some information here, but not in a way that actually matters in practice. So I'm going to copy that. I'm going to search. And so we see that there really aren't any shared features between these. So it looks like donor and genotype are completely independent effects. Okay. So the next question is, how many differentially abundant features are there between the two genotypes? And are they more abundant in the wild type or susceptible? Fortunately, the genotype's the simple one, and we kind of went over this. So for the most part, it seems that the wild type has the higher abundance, because these two were just differentially present. This one was differentially abundant, and we can also see that the effect of this was 2.4. So I don't like thinking about log odds, which is kind of what this is, so I'm not going to stew on that too much, but we can see it's a pretty good effect, and it's positive. So we have more in the comparison of susceptible against wild type. Okay, and the last question, use taxonomy metadata visualization and search sequence identifiers for the significantly different features by genotype. What genera do they belong to? So I'm actually going to leave that as an exercise for you guys, because it's important to be able to kind of think about these things, but I will give you a hint. So the taxonomy metadata visualization, we generated up here a little bit. So we have our taxonomy bar chart, and over here we have a tabulate seeks. Or sorry, not tabulate seeks, tabulate metadata. So here we're using the taxonomy file as metadata and producing that visualization. So I'm just going to look at that. And so you can imagine you can actually search those differentially abundant features, and you could look at these and try and understand, is there any kind of like taxonomic interpretation for these features? So basically, do we interpret these as amplicon sequence variants, or is there some more general pattern we can identify using taxonomy? So that's basically all there is to it. Thank you for listening, and hopefully this was helpful.
Welcome back. Thank you, Evan, for that tutorial walkthrough. I'm going to give Evan just a moment so that he can switch over into the other chat here. And while he is doing that to take questions, I'm going to give a couple of brief announcements here. First off, before we get into questions and then into the general question and answer session for the end of the day, I just want to point out something with the schedule here, and that is this upcoming general Q&A session will be the last scheduled general Q&A for the workshop. So if you have general questions that you want to send our direction, please think about those as we're talking here this afternoon and get them into the queue. We will try to fill in more general question and answer tomorrow if we have some downtime between sections, but it will kind of depend on the nature of the questions. But our last scheduled time is coming up here in about 30 minutes. So it looks like Evan joined us here. So Evan, I've got some questions for you. So the first one in the queue here is someone would like to know, is it possible to run ANCOM BC in QIIME 2? Or if not, are there any future plans?
So it's not possible just yet, and I'm getting some feedback. Oh, I understand why. One moment. I have way too many audio devices to monitor the stream, and also to be here. It gets really confusing. So it is not present yet, but there's actually work happening like literally right now to kind of make that happen. There's a few emails in my inbox I need to probably read related to this. So look for it in the near future. Jamie might have some more things to say about this.
Oh, sorry, I didn't realize that's not a queue, is it?
It was.
Okay.
What can you tell us about ANCOM BC?
Yeah, so it's actually really cool. So ANCOM BC is a bit different from the previous version of ANCOM. It returns something that looks more like ALDEX2 and Songbird in the sense that you can get the logful changes directly from it. But the one thing that ANCOM BC does is it under some assumptions, it tries to correct for the biases induced from the absolute abundances. As we mentioned in the previous lecture, that the differential abundance has issues because of the change in absolute abundances we can't measure. And ANCOM BC, under a few additional assumptions, it can try to correct for this change in absolute abundance.
Yeah, I'm really excited about
features looking great. I've got another question for you here, Evan. How can I work with my taxonomic classification, taxonomic identifications in the differential abundance step that we just covered in ANCOM?
Right. So this question was asked just before the section where we actually did that. But there are actually a few ways to handle this. So suppose you are doing some kind of meta-analysis where something like closed reference or taxonomic-based features are more interesting. You could certainly collapse your features by some kind of taxonomic level and then do ANCOM on that feature table. But probably the best first approach is to kind of do what we did in the video, where you take those ASVs and you can look up the taxonomy based on that.
That's great, thank you. Let's see, last question that's in the queue here is related to filtering features. We had a filtering step there, and the question is how can we determine an appropriate minimum frequency for filtering features?
Yeah, I don't have a good answer to that. Justine, do you have a better sense of how to think about that?
Sure. So I have many opinions on this topic. The control room is laughing because I have many opinions on many topics. So I tend to like to do it's hard to do right now in QIIME 2, but I tend to like to do a prevalence-based filter or a joint prevalence abundance-based filter. And so for my prevalence-based filter, I like to keep features that are present in about 10% of my samples. I think it gives me sort of the best behavior empirically when I'm working with prevalence-based things. And it also tends to collapse my list of features pretty significantly. So I'm only getting probably the top 10%. And if you're doing FDR correction and you go from 3,000 to 300 on features that are present in, one or two samples, you suddenly have a lot more power to do your detection, and you have thrown out a bunch of crap that you're going to have to penalize for. And so here we had 48 samples, and I think we kept things that were present in 4 samples, which would be right around that 10% threshold. In terms of relative abundance, or absolute abundance, you may also want to filter on counts on the off chance that there's something that's like 1 count across 100 samples, and then would have 100 counts. That tends to be less common, but is another place to filter. But yeah, no, for me, it's prevalence filtering, and it's about a 10% threshold.
Thanks, Justine. Yeah, that's really helpful. I'm wondering, does anyone else in the control room here have any questions for Evan or for Jamie about differential abundance or about this section of the ANCOM tutorial? All right. Well, if you think of anything, or if any of the attendees on Slack think of something, just send your question in on general, and we will we'll see that and get that into our queue. So I think with that, it's time to move into our last general question and answer session. Does that sound good? Okay, I am going to pull up my list. I think we've got a nice list populated here. So I think this first question that I have here is for Ariel. Are you around? Welcome.
I'm around. I'm not sure I'm the best one to answer this question. My little thing is there because I replied to them, but
I see.
This is not the one. But I mean, this is the PCOA and PCA one, right?
Well, then I'll ask you the one that you had mentioned before. So control room, we're going to ask the third question down in our list here. What will be the basic differences between unifrac, Jacquard, Brake-Curtis distances? And I'm going to hand that over to Ariel. So this is a question from earlier today that's in our queue.
Yes. So, I assume the reason that you were asking this question is specifically related to those beta group significance tests and not like what those metrics are, but just in case, I'm going to just very briefly touch on it. And if you do want the details, you should have access to the slides from yesterday that have like the mathematical formulas for these in them. So hopefully that will help you. But right, the big difference here is unifrac is phylogenetic, right? So it takes into account the evolutionary relatedness between SVs in your sample, whereas Jacquard and Brake-Curtis are not. And they have, right, one of them, or Jacquard is just richness, and then Brake-Curtis takes into account evenness. So that's like the main difference, right? I kind of like to think about them if those alpha diversity metrics were easier for you to understand because they're a little simpler. I like to line them up, right, where you have Jacquard like observed OTUs, Brake-Curtis like Shannon's, and unifrac like the Fates phylogenetic, right? I didn't go those backwards, did I? Okay, great. Sometimes I do. Anyway, so that's like the major difference between those, right? And because they're different mathematically, when you're doing those statistical comparisons, it's telling you different things, right? If you have 2 groups in your samples that are the same, they are not statistically different with Jacquard and Brake-Curtis, but they are with unifrac, that can tell you that maybe they learn numbers of organisms in them that are equally distributed, but they're from 2 different clades of the phylogenetic tree, so they're actually pretty different, something like that. So that's like the rough answer for this. And so kind of going back to some things I think we've already mentioned a couple times now, where you can you can show more than one of these in your paper. You should talk about more than one of these in your discussion because they do tell you distinctly different things about your data. They're all valuable, and not getting significant in one doesn't mean that something's wrong with that metric. So I hope that helps. If I misunderstood your question, you can reply in the thread there and we can come back to it or something.
Thank you. Yeah, that's a really important reminder about considering sort of the whole picture there. I saw Mike had jumped on. Did you have something that you wanted to add to that discussion? No? Okay. So then I'm going to move on to the next question here. So backing up a little bit into this morning's tutorial section, can someone explain the difference between PCOA and PCA? And I think I'm going to ask Evan to help with that.
All right. This is pretty similar to a question yesterday. And so the difference is basically the metric you're using. So in PCA, you're using Euclidean distance, whereas in PCOA, or principal coordinates analysis, you are using kind of any distance space, so any metric space, sorry. In fact, if you're to do principal coordinates analysis on Euclidean distance, so you create a distance matrix, but it's Euclidean distance instead of something like Ray-Curtis or Unifrac, you would end up with exactly PCA. Now, using Euclidean distance in microbiome data is probably not the best approach because the data is definitely not Euclidean, but related to the compositional stuff we've been talking about, if we do something like a CLR transform or an ALR, you can actually do a Euclidean distance. And so that's what we call Atchison distance, and we have that implemented in QIIME 2 as well. You can also do a robust Atchison ordination using Decode, which is a pretty cool plugin.
Great, thanks, Evan. Let's see, my next question here. Let's see, I think I'm going to put Bod on the spot, and then you can hand it off if you'd like. So this next question in the list here, going back to, I believe, our beta group significance discussion this morning, what would it mean if variance was higher in one group versus the other? I.e., if perm-dis was significant, how would I interpret that result?
Yeah, so just to, again, clarify the difference between these two, the perma-dispersion is, as the name suggests, it's checking whether the variance within the groups are different across from each other, whereas the permanova is looking at the within group differences across to across group differences or variance. So, and I think this was covered in the videos that one should be only taken, the perma-dis should be run alongside with the permanova to sort of check one of the main assumptions of the permanova, which means to say that the variance between of each group is similar to each other. So if you get into a situation where that's not the case, so permanova says there is a difference between the groups, but the perma-dis also says that. So that suggests that there might be something underlying the actual structure of the data set which you should be aware of and interpret those results very carefully. That's not to say that there isn't a difference between the groups as the permanova suggests, but the fact that is that you haven't met the assumption of the test, and then that you should be very careful with the interpretation that there might be something else driving that the results. One of the things that leads into that oftentimes is if you have unequal sample sizes. So as you imagine, as sample size increase, usually tends to increase the variance as well. So that's one of those factors that's actually quite sensitive with a test like permanova. So if you can manage to increase your sample sizes so they're even, that's usually one way to sort of alleviate that. But yeah, it's definitely a sort of a red flag in a sense that you should be aware, but doesn't necessarily mean that the opposite of what you think is also true.
Great, thanks, Bod. I think I want to see if Chris is available for a question about our Emperor plot.
I am available.
Hello, Chris. The question we have here is, in our principal coordinates plot, do we need to ignore the percent variance in the axes, or what should we be doing with this information?
I'm not actually sure what that question means. I would love to answer, but I think I'm going to have to pass this off.
I can maybe help clarify the question for you because I saw it when it came in. And what I think he's asking, we were talking about how you can't get any sort of statistical significance from the PCOA plot. So does that mean those numbers don't matter? Does that help you?
Yeah, are we talking, I'm going to assume that we're talking about the numbers that are kind of displayed at the end of the axes. And so though we can't use that plot to kind of quantify statistical significance, those numbers are still useful because they explain the percentage of difference that is explained by the axis itself. So if you axis 1 with 35% at the end, it means that 35% of the overall difference in your selected distance metric is explained by axis 1. And that can be really useful in kind of deciding which axes you care about. If you have a really strong value for difference explained by axis 1, you probably care about change in axis 1. Whereas by the time you get to axis 5 or 6, if the percentage explained is very small, you probably don't care as much about that axis.
Can I add on just a little bit to that?
Please.
Right. So I think just to make a basic understanding of these PCOA plots, right? Because a lot of times people want to know what those axes represent, and it's not representing anything in your metadata unless you force it, right? So it's like Chris said, it's like the difference in those samples. But I think the big thing in interpreting it, sometimes you'll see data and it looks like there's a huge difference between these two groups. They're all the way across the axis, and then you look and it only explains 7% of your data. And so you say, oh, maybe that difference isn't as big as I think. But I think for you in your analysis, so that's good to know if you're reading a paper. But something for your own analysis is you can look at that and think about what variables you might be missing or what's not happening in your data that you don't get that trend you expected as much, if that makes sense. There might be other things in there that are explaining it if only 5% is explained by those metadata differences.
I think it's also probably worth noting or mentioning that as the number of samples goes up, the variance that is explained by each individual PC will go down. And so if you have 48 samples, you might explain 50% of your data with the first PC. But if you have, 4,800 samples, you might only explain 5% of your variation with that PC just because you've got more information that's packing the back set of PCs. And so you have less weighting on the first one, even though it's still the most amount of variation in your samples.
Thank you, Justine.
Sorry.
No, go ahead.
There is a little graph under that axis tab on the Emperor thing that tells you those distributions if you want to see that.
Thank you. Yeah, that's a good reminder. Let's move on to the next question here. Justine, I'm going to send this your direction. Why are we using non-parametric tests to test data with a normal distribution? And there's a reminder in here. Yesterday it was mentioned that the Parkinson's mouse has a normal distribution.
Okay, so there are a couple pieces here, right? We have this. So yesterday we talked about alpha diversity and about how we assume alpha diversity. So the general assumption with alpha diversity, the historic assumption has been that it is non-normal. And so we use a non-normal test for non-normal data. You can still use that non-normal test for normal data. You're not violating assumptions, but you might be losing some power. However, there is this fun little principle where a lot of your richness metrics are at least observationally asymptotically normal. And then I see this also involves the PermaNova test. So the reason we use the PermaNova test for distances as a non-parametric test is that we have this problem in distances that I think Ariel presented, that a distance is between 2 points. Am I reading the wrong question, Matt?
No,
okay.
No, I think you're good. Thank you.
Okay, so a distance is between 2 points, right? And we have the triangle inequality that says there's always sort of a third point in there, and so the distance between A and B and A and C is also dependent on the distance between B and C. And one of the major assumptions of most parametric tests, and actually most classical statistical tests, is that the data is independently distributed. And so we break that independence. And so the permutive test as a non-parametric test lets us deal with the fact that we're violating this big assumption that we normally have in our traditional tests. And so that's why we approach it with Permanova here. Do others have thoughts? Because I'm happy to have other people have thoughts here too.
No comments here. Okay, thanks, Justine. So let's see. Greg, I'd like to ask a question to you, if you're available. So
Yep.
I'm looking at the one that you maybe accidentally deleted out of the general channel.
Right.
So
yeah, let me pull that one up.
So I believe the question is it possible to filter features based on their percent abundance or relative abundance rather than the overall frequency?
Right. Yeah, that's a good question. I don't remember the answer to that one offhand. And so we would use QIIME feature table filter features for that. But I think we have a abundance option in there, but I'm not certain. I can double check on that if we want to come back to it in just a second, unless anybody knows offhand.
I don't know offhand. I seem to recall though that we don't have that option, but I'm not 100% sure. But yeah, we can circle back on it.
Yeah, let me check on that, and I will follow up in just a second.
Let's see. I will look at my queue. Oh,
yeah. So we do not currently have that support, and so yeah, unfortunately we don't. It's probably something that we should get in there.
There are requests for that already in place as well.
Cool. Okay.
I think I might even have a PR for it in.
Oh yeah. Okay.
That's right. Yeah, it's on the to-do list to take a look at that. Thanks, Justine.
Yeah, so when we're talking about like requests and PRs here, we're kind of talking about what we have going on GitHub. And so that would be an issue that somebody opened for a feature request on GitHub. And then a PR would be a pull request. So Justine thinks she may have actually submitted some code that would add that functionality to the plugin. And so, yeah, we'll definitely check on that.
All right. Let's see. What do I have here? Is it wrong to carry out the step of assigning taxonomies before carrying out the downstream analyses in order to be able to run all the analyses at different levels? Maybe different levels can highlight different behaviors of the data. Mike, is that something you'd like to talk about?
Yeah, I can chime in with a few thoughts unless somebody else has.
Can you move your mic down?
Okay. Sorry, I keep forgetting to have this mic thing. Yeah, so I typically I recommend folks classify their sequences prior to any downstream analysis, and there's a few reasons for that. One is especially those of you that might do 16S sequencing on plants or animals, whatever, you'll have a lot of a lot of the primer sets will amplify the 16S gene from chloroplast or mitochondria, right? So those are not really indicative of the microbiome you're trying to analyze. So you want to try to remove those features from your data set, because if you have a high abundant of, in a sense, contaminants, plant contaminants in your data, that's going to affect your diversity metrics as you move through everything. So I try to filter those. Now, there's a lot of variation on how you can filter and what you can think of when you want to filter based on taxonomy. Usually, I remove all plastid sequences, so your mitochondria, your chloroplast. I also quite often remove anything that does not have at least a phylum level description. But that varies based on your data, based on the questions you're after. But for me, that's generally what I do. I also typically remove unclassified data because most of my work, I'm not interested in discovering novel taxa. So that's something you also may want to leave in or not, depending on your question. Now, there are some other ways within, the QIIME universe to maybe make more coarse evaluations of your data. There are ways you can merge your different features based on taxonomy. And again, this could be dependent on if you're looking at genus level or family level patterns in the data, depending on your question. That's in a nutshell on how I look at it, but I'm sure others have thoughts.
I think this participant had a few more kind of follow-up questions in the conversation on Slack. So I'm curious to get your opinion on using something like down to like a specific tax, like if you collapse down to a specific taxonomy level and use that for like your diversity analyses. What are your thoughts on that? And I know Justine has thoughts too, so.
Well, so as we've been, the recurring theme throughout the workshop is you should always try multiple things, multiple ways in your data, and that's a very good way to sanity check any patterns. So if you're finding, say, a certain family of organisms that's enriched in a group or more prone to be in some treatment group, just using your feature data, hope that should also come out in the wash when you collapse your data. But sometimes, depending on how good your taxonomy assignment is, or maybe how odd the sequence sampling is among your samples, you might have a lot of, say, taxa of the same genus, or sorry, different genera, but they're all within the same family, and you want to get, you can look at that. But sometimes I try to do multi-level approaches for that because, again, for a lot of these data sets, I'm not sure what the pattern really is, and this is the whole point of investigating. So don't be too, don't limit yourself to thinking you can only do an analysis one way and then whatever that answer is. So I try to do a multi-tiered approach on how I collapse, but I'll defer to somebody else that may have a better description on how to do that.
I just had one comment to add here, going back to sort of the original question, which was, I guess, about the order in which things to be done. I just want to clarify that what Mike suggests is something I agree with, that, getting your taxonomic assignments is a great idea to do it at the very beginning, because as he said, there are uses for it. For example, filtering your mitochondria, chloroplast, or other unknown or unassigned values. But that doesn't mean that you still can't do, analysis at the ASV level. What you have is essentially an artifact that you have taxonomic assignment. It's just another way of looking at your data. So when you need it, you call it, but you could still do your analysis without it at the ASV level. Or if you want to look at patterns at the genus level, then you can bring in that file and say, okay, now I want you to collapse these at the genus level and look at that. So it shouldn't be really thought of as, whether you can do this or that. Is that you have the option of doing all, and the taxonomy, that's why it's actually, kept in a separate artifact altogether, is that you can call it for the specific question that you have.
I'd like to add to that. Bod reminded me of something. I've come across a few papers that have been published microbiome data, and one thing I would like to point out as a public service announcement with taxonomy is keep in mind that chloroplasts and mitochondria, the upper level taxonomic descriptions like the phylum, class, or family level, are actually taxonomic descriptors of the bacterium they evolved from. So cyanobacteria is actually what the chloroplast is within. So be very careful when you're making box plots or taxonomy plots based on phylum level. There's a few cases I caught where folks thought they had a lot of cyanobacteria in the data, but when you actually look at the complete taxonomy or even do a tree, you'll see that they were actually contaminant plant sequences. And I think for mitochondria, hopefully I'm getting this right, I think it's alpha proteobacteria that mitochondria fall under. So that is the most common taxonomic issue that I find. So when you're looking only at phylum level, make sure you look a little deeper and make sure nothing else is, I think it's class or family for that describe whether or not it's a chloroplast or mitochondria.
Can I just make a general appeal that people stop doing phylum level analysis? Like, this is my public appeal to you. Phylum level analysis is super, super high up. Everything with a spinal cord is in the same phylum. And as an ecological niche, I agree that the ratio of things with spinal cords to the skeletons may inform you of something, for example, your risk of global warming. However, it turns out that if you're looking for a risk of global warming, there is in fact a specific species and possibly even a subspecies that explains a lot of current climate change. Yes, that may propagate all the way up to the phylum level, but in reality, it's much more interesting lower down taxonomically. This is like my appeal to you as you collapse your data. I think family level can tell you a lot. I think genus level can tell you a lot. I'm a little iffy on species and 16S. I'm totally into ASVs. But please, 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 no more phylum level collapsing.
Thanks for that, Justine. Mike, I've got a question for you here. Which one can give us more information at the species level, the Silva database or the Green Genes database?
Yes. I have lots of opinions on taxonomy. So step one, be wary when you use when we're asking or discussing species. So I put a response in that thread, so it's hard for me to disambiguate if you're talking about just the number of reference sequences in the database, or if you're actually talking about species descriptors. Now, as far as I know, Silva, for example, has a lot more data in the database compared to Green Genes. However, they don't necessarily curate the species label fields. So technically, one could argue they only go down to genus. However, through a new plugin that recently came out, we allow an ability, and actually in the QIIME classifiers we provide for Silva, we have a way of appending organism names to that species fields, but there's a lot of issues with that. A lot of them are meaningless. They'll just say metagenome or unclassified bacillus. So be very wary in general with amplicon data of believing species labels. Oftentimes, depending on the limitation of a database, so with my experience, at least for the data I've worked with, now everybody's story is going to be quite different. With the Green Genes, for example, there are certain groups that I get overclassification, meaning it's saying that this particular taxonomic descriptor is of this species, but really there's only a pool of those related organisms that are in that database, so it keeps hitting that, or the reads keep hitting that set of taxa, and they all happen to have the same species labels, so it's going to call it species X, when really it's probably a different species. So keep that in mind when you're using a lot of these different taxonomy databases, whether they be Green Genes, Silva, GTDB, and there's a variety of others out there that you can use. But always be wary and don't be afraid to compare and contrast the different taxonomic signal that you get, because you might find that one is going to always call an organism one thing and another another. And a lot of that really is a problem of annotation, right? Humans involved in annotating this data. So I know this is a long-winded answer to your question, but I will just say that Silva and I believe maybe GTDB have more reference sequences than Green Genes. However, Green Genes might have more relevant taxa for, say, human microbiome. So you just kind of want to make sure what those reference databases are optimized for.
Thanks for that. So the next question in the queue here is about the taxonomy bar plots that came up before. And the question is it possible to group our samples based on some metadata group? I'll go ahead and answer that one briefly. It is technically possible, but it's requires a little bit of gymnastics in order to make that happen right now. We have some open feature requests, like what Greg was talking about earlier, about how we kind of keep track of the things we want to do. We have some feature requests about new ways to visualize and regroup these plots on the fly. So otherwise, if that's something you wanted to do today, we would suggest that you export the data and then plot it in the tool of your choice.
Can I add to that?
Yes, go ahead.
There is to use the QIIME feature table group command. No,
not easily, not without some gymnastics there. So it would require you to rebuild your metadata.
Got it. Okay. Yeah. And actually what I was going to say, which I realize now that's not possible, but the downside to doing something like that would be that if you had, well, first of all, like you wouldn't necessarily see how many samples were making up each group. And then you'd be doing some sort of an average across those. And it's possible that an outlier could really throw off what you think the composition is of those groups. So I would just tend to be pretty careful with that. Like I would rather see the samples grouped together like you can do in the taxa bar plot right now, because then you can sort of mentally look at that and figure out, okay, these ones seem to have a higher abundance of proteobacteria or whatever, but at least you get to see each data point when you do that.
Yeah, thank you. That's a really good point. For my next question, I want to put Andrew in the spotlight here. Andrew, are you on the call?
Oh dear, what is it, Matt?
Hello, Andrew. We have a question here about QIIME 2 Studio. What's the best way or the recommended way for running QIIME 2 on a Windows computer?
That's a good question. So first of all, QIIME 2 Studio is not exactly where we're focusing our efforts anymore. We might provide, we're interested in providing a similar solution that is much better quality than QIIME 2 Studio. QIIME 2 Studio is not necessarily our most well-engineered product. It's something that we don't put a lot of energy into anymore. We've discussed some new and improved versions of it. The best way, what was the rest of the question? The best way to run it on a Windows computer?
Yeah, how could I go ahead and run this on a Windows computer today?
Oh my gosh, I'm totally blanking. It's been a long time since I played with QIIME 2 Studio.
So, well, I can hand the rest of the question over to Evan here. Thanks,
Please
Andrew.
take it from here.
Hello. So good question. This is something that definitely comes up a lot because there is definitely demand for a graphical interface, right? And so Andrew's right. Q2 Studio is really not kind of where we want it to be. It's really a prototype of kind of this idea of the QIIME 2 architecture allowing us to generate these interfaces automatically. And so QIIME 2 Studio is really just an example of it is in principle possible to generate a graphical interface dynamically. That said, it is sometimes still nice to use. So if I were on Windows, probably the most straightforward way to do this would be to use a VirtualBox, because that has Q2 Studio already installed in it. What I would really like to see in the future is some kind of system where we actually connect to the Windows subsystem for Linux. And so then some native Windows application that basically calls through that layer. And so you could imagine doing all of your analyses in a pretty standard Windows application. That is a pretty massive undertaking, and so we haven't really gone in that direction yet. What we're working on instead is this Galaxy interface, which is a really powerful and well-established graphical interface tool for bioinformatics. And so just like we can generate command line interfaces, programmatic interfaces, graphical interfaces, we can generate Galaxy wrappers as well. So our goal is to kind of basically make it QIIME 2 sit wherever it needs to be to make the end users most comfortable. And I'm just checking. Yeah, so definitely no installer yet. Everything's source install. So yeah, Q2 Studio's kind of been put on the bench, so to say. And we're kind of looking at Q2 Galaxy as like the most immediate and direct way to get a graphical interface out for QIIME 2.
Thanks, Evan. Yeah, I think one thing to add on to that as well is there's no requirement that you conduct all steps of an analysis in any one particular interface in QIIME 2 as well. So you could, start and do some of the more CPU intensive or memory intensive steps using Q2 CLI on your university's compute cluster, and then move over for some of the downstream steps into the QIIME 2 Studio interface and work on it in that interface, and then maybe switch over back to the Q2 CLI or something else. Just want to point out we're not locked into a particular interface for an entire analysis.
But Matt, is my provenance tracked if I do that?
Thanks, Evan. That's a great question. Yes, your provenance is tracked if you do that. So QIIME 2 is going to be keeping track of what you did in all of your steps that you've run through various QIIME 2 plugins or import steps, no matter which interface you're working in. Let's see. Next question here. The question is about, in QIIME 1, there was a script, split libraries fastq.py, and there was a parameter in there where you could specify the maximum number of barcode errors. And this user has pointed out that the QIIME demux emp paired command doesn't have that option. We can either just enable Golay error correction or disable it. And I wanted to just comment on that briefly, and then if anyone wants to follow up, you're welcome to jump in. So with the EMP paired demultiplexing step, you're correct. There is no way to currently parameterize or specify the maximum number of errors in the barcodes there. Basically, this plugin is, if Golay error correction is on, it is going to attempt to completely correct the barcode. I think I was looking into the source code for this a little bit, and I think it can correct up to 3 bits of error in there, and then otherwise, if it's not able to correct the barcode, it's just going to say, I don't know what sample this read belongs to. So that discussion there is specific to the EMP paired command in the Q2 demux plugin. I'm also pretty familiar with the Q2 Cut Adapt plugin as well, and this is for used primarily for demultiplexing sequencing product that has the barcodes still in the sequence, so they're not EMP formatted where you have a separate index barcode file, but you have the barcodes are actually in your reads. There, in that plugin, there is the ability to kind of specify the tolerable number of errors, or there's also like ways to configure the indels or substitution errors. There's a couple of different ways to kind of calibrate that based on your needs. Anyone want to add anything to that? Okay. Let's see, my next question here. How should we interpret a feature with S underscore underscore found to be differentially abundant? I see some thinking faces up there.
I'm happy to take that one, unless anybody else wants to. Yeah, so you would interpret this the same way that you would interpret any other feature being differentially abundant. It just has this caveat that the database doesn't have an annotation for that sequence. And so when you see that S underscore underscore, that basically means that that sequence, there's a close match in the database, but the creators of the database didn't know how to annotate that sequence. So you can, so I guess another question there is like, well, what can you do with it if you're interested in that feature? One thing that you could do is you could take that feature ID, and so that would be that big ASV ID. You could look up the actual sequence for that, and we looked at a visualization earlier in the workshop. It was the representative sequences, where you could see what the sequence was that was associated with each of those ASV IDs. You could use that interface to blast the sequence against NCBI, and you could get NCBI's take on what that sequence is. But just note that if you are going to do that, make sure that you're not just looking at the first blast result that you get back. You really want to look at maybe the first 10 or 20 sequences, and you want to just see, do these have a consistent annotation, or is there some variation here? There's probably a reason why that sequence is not well annotated in the database, and it might be because there's just some ambiguity, or it's just unknown what, or maybe an unnamed species that sequence has been isolated from.
Thanks, Greg. Ariel, did you have something you wanted to add to that?
I can, yeah. It's funny because I ran into issues with this labeling before and had to ask about it in the forum. So I'm there with you all. But one thing that I think is important to note is like sometimes if you really closely look at how everything annotated, you'll see like they go down to the same genus, right? And then one of them has that S underscore underscore and one of them doesn't. It just stops at the genus. And I just want to point out the difference between those, because the ones that stop at genus, they couldn't like distinguish it down to the species level at all. So you just know it's from that genus. Whereas if it has the S underscore underscore, like Greg said, we know it aligns with a specific species. We just don't have like a name for it. So that's why those show up differently, even though it seems like they should be the same thing, which is what I had to ask.
Thank you. Mike, did you have something you wanted to add there? Or? No? Okay. Jamie, if you're still around, I've got a question for you.
Shoot.
Why would I pick ANCOM or Songbird for different analyses or different questions? Like, what are some of the criteria that I should be thinking about when picking a tool like ANCOM or Songbird?
Well, I think it really boils down to what assumptions are you willing to make for your tools. Now, I do want to point out that the old version of ANCOM is very different from the previous, the other differential abundance tools, because namely ANCOM is looking at all pairwise comparisons and trying to see which microbe really sticks out of the crowd. If that's the kind of thing you want to look for, if you want to see which microbe contrasts most heavily with all the other ones, and you are really bent on identifying individual microbes, then yeah, by all means, that's fine. But I want to point out that the other differential abundance tools like Songbird, ALDEX2, ANCOM BC, all of those give you something that looks like a log4 change. But I do want to point out that each one of them handles statistical testing differently. So in the sense that we know that we can identify which microbes have changed, but we can only do it up to a proportion because we don't know how much the change in absolute abundance is. So in order to do a statistical test, every tool has to make some assumption. For instance, with ALDEX2, with DSeq2, they do is they assume that the average microbe isn't changing, which basically means that you're assuming that there isn't a change in biomass. So if you have a growth in the entire microbial community, that assumption is violated. So with Songbird, we don't make this assumption, but at a cost that you can't do statistical testing. So the main difference, so in summary, the main difference between all these tools is that every tool has slightly different modeling assumptions, but most of the tools, particularly the recent differential abundance tools, will give you an ordered list of microbes, ordered by how much they're expected to change.
Great, thank you. Evan, are you around for an ANCOM question? Could you go over the meaning of the W statistic again?
Yeah, so if we recall, the thing we can compare between samples is really a log ratio. So we set up some kind of simple ANOVA where we had a log ratio for maybe sample 1 and a log ratio for sample 2. And we had that formula where we had an intercept, some additive effect of that sample group, and then some error. And so what we were asking was, are these two additive effects different? Or maybe you have n of them. It doesn't really matter. But are the betas, the effect of that group, significantly different? And so there's a few ways you can actually decide what kind of statistical test you want. But the W is concerned with how many of those comparisons were found to be significant under your framework. So the W is really, if you think about that graph again, where we had all the pairwise comparisons, it's the number of edges that were significant. So it's just the number of significant log ratios. Hopefully that's helpful.
That's great, thank you. Let's see. My next question in the queue here, it's an opinion question. So I'm gonna maybe just tweak it just a little bit and see, anyone on the call here have any experience with using PyCrust2, the Q2 PyCrust2 plugin for doing metagenomic prediction? Anyone care to chat about that for a minute? Just talk about your thoughts or opinions there.
I've just 1 and 2 with some success in some capacities. It's of course it's one of these tools where the whole purpose behind it is to extract as much as data as you can from your existing data. It's not meant to be used as an absolute thing. If anything, at most you should be using it to generate hypotheses that you can test with real data. And with that, I found it useful in the past, and I've had publications that benefited from using it. PyCrust2 is definitely a huge improvement over PyCrust1. It allows you to use the ASVs, and it doesn't have a lot of the limitations that PyCrust1 did. So if you're going to use them, I think they're a great tool. They're well designed and all that. But just be aware of there are a lot of limitations. Actually, the authors do a fantastic job of listing all the limitations for that tool in their own website. So my advice would be to make sure you read those very carefully. Definitely know the assumptions, the limitations, and then apply it to your tool after. Don't just sort of go looking for something and then try to validate it later. Have sort of prior going into that. But yeah, it's I think it's a fantastic tool for what it can, what it's designed for.
Thank you for sharing. This one might be another question for Evan here. Question is, why are Euclidean distance matrices not recommended for microbiome studies? I think maybe we're asking about these metrics there, but
yeah. So that's a good question, and I think one way to kind of think about it intuitively is if you think about what the metric, or distance in this case, is telling us. So if we calculate a Euclidean distance between two samples, we are treating basically every single feature has a totally independent orthogonal axis, right? So if we imagine 3 features, you could think of a 3D plot, where you have your 3 axes and you're moving in some direction on each of them, and that'll give you a little point in space. And then we're going to measure against another sample of 3 features a distance to that. And if you think about biology, that's maybe not the most meaningful kind of result, because it's really saying that you can square all the components, add them together, and take the square root. I think I got that right. Correct me if I'm wrong. I'm pretty sure that's Euclidean distance. And there's really no reason to expect that to be true. That being said, there's not always great justifications to expect many distance metrics to be true. But they're often a little bit more, they're less strict, I guess, is the way I would put it, in terms of how they think about distances between points. And so it allows for the space that you're looking at to kind of be not a very strong, rigid lattice of points, but rather like A and B are pretty close to each other. A and C can also be pretty close to each other. And we're going to make a statement about A and C together, but it doesn't have to be like the square root of something squared. We're going to just have the triangle inequality, which is much less difficult to achieve, and probably fits more in line with what we might expect with biology, where we would have, we really don't expect our features to be independent, right? We expect there to be some kind of ecosystem happening here. We expect there to be one bacteria is causing another bacteria to either increase in abundance or decrease. There's all kinds of ecological effects we're interested in. And so treating every single ASV or OTU as an orthogonal axis where you can like perfectly cut a distance between them doesn't really make a lot of sense, I think.
Cool, thank you. Greg, did you have something you wanted to add to that?
I guess, yeah, the only thing that I would add to that is, if you're interested in experimenting with that, you can compute a Euclidean distance with QIIME diversity beta. And if you generate a PCOA from that, anytime I've ever done that, you can just tell looking at that PCOA that something is wrong. Like that data just doesn't look right. And so this might be something like all of your samples grouped together in one quarter, except for one sample, which is way out on the side. But you can, if you want to experiment with that, take a look and you'll see what it looks like to get a really funky looking ordination plot.
I threw a paper into the chat. That's an older paper where they compared like several metrics, including Euclidean distance and unifrac. So that may also be worth looking at if you don't want to mess around with your own data to do what Greg suggested.
Cool, thank you. Let's see. We've got a question in the queue here that I think it's come up a couple of times now, but I think we're going to revisit it again since there seems to be some lingering questions about it. And this has to do with when we should rarefy our tables or not, our feature tables. So there's a question about is it okay to skip rarefying my taxonomy table in order to investigate low abundance microorganisms? And perhaps we're also kind of thinking about the low feature abundance filtering at this step too. I'm not sure the context of when this question came in, but would someone like to comment on that?
I can take a whack at it, and then, or Bod can. Jamie, I don't know if one of you guys want to take it.
Go for it, Justine.
Okay. So, I think the conclusion sort of was it Tuesday that we talked about rarefaction? Tuesday, Wednesday, as we talked about rarefaction with diversity metrics, because they are sensitive to your sequencing depth. And there is a paper that if we haven't shared, I will share that talks a little bit more about that. But your diversity metrics are sensitive to your sequencing depth. So if you don't rarefy in diversity, one of the major components that you will observe is that effect of sequencing depth. Diversity captures all of your microbes, your rare microbes, your common microbes. It sort of picks all of that up because a rare microbe theoretically has the same probability of being drawn out of the hat, whether, like, given your probability distribution when you rarefy to a fixed depth. The other piece, though, is I talked about filtering rare microbes earlier, that I like to filter things that are present in less than 10% of my population. And basically that's a power approach, because if it's only present in one person, it doesn't matter how awesome it is. It could be the one microbe that saves us all. If there's only one microbe that's present, I can't do anything with it statistically, and that makes it really hard for me to work with. There are other people who do like microbe hunting and go out and look for the most interesting, unique thing they can find, and they're trying to capture all the diversity. And their perspective on this may be really different than mine because they're cataloging and I'm looking for things that are differentially abundant. So can I go back to the CHIME workshop theme? It depends. And I think others probably have strong opinions. I'm sure Jamie has opinions here, so I'll hand it off to him.
Thanks, Justine. I think it also depends on the metric you're using. There are some metrics that handle this more gracefully than others. But I will point out that I agree with Justine. I think a lot of it boils down to what are you willing to do with uneven sequencing depths. Now, there are some metrics that will attempt to put a lower confidence on the samples that have smaller sequencing depths. And tools like that use the multinomial distribution are very apt at doing this. When you have a sample with a smaller sequencing depth, you have a greater uncertainty about what the proportions of the microbes actually are. So most of the differential abundance tools can handle uneven sequencing depths, like ANCOM, ALDEX2, Songbird, and DSeq2. But I think that this front still has a bit more work to do to fully answer this question.
Thank you. Anyone else want to comment on that?
I think it was beautifully said, and as always, it depends, comes back. When you're filtering based on the number of features, you should also be aware that there's features that are present across different samples too, so different dimensions and layers of filtering. So for tools like ANCOM, Songbird, and stuff, if you're comparing, for example, a type of bacteria that's really abundant in one of your groups and not at all in the other ones, by default you're going to identify that as something that's abundant. But if you're working with really rare features that happen only 2 or 3 times in one group and in one sample in the other, then those aren't really super informative features. So as Justine was talking about, is that including those, not only does it not really add any information, like you can't really infer anything from it, but it also adds a lot of noise in the data. So with that regards, it's tool specific. If you're using differential abundance, then that kind of filtering might be useful. But for those broader picture beta diversities done, those are actually quite informative, and they can also be quite influential in the model. Like they can really drastically change the outcome. So yeah.
Thanks for that. My next question is directed at Greg. Greg, is there a standard operating protocol for analyzing 16S rRNA data using QIIME2?
That is a very good question. So, in our tutorials online, like the PD mouse tutorial that we're working through here, or the moving pictures tutorial, we go through, what are kind of like standard operating protocols. But I usually sort of tend to think of these as kind of like, rather than an SOP, I usually kind of think of it as my first pass at an analysis. Like these are usually the things that I do when I'm first trying to get an initial look at the data. And then every study is going to end up being different. The goals of the study are going to be different. And so like we've been saying, what you want to do depends on the goals of your study. If you, there may be, some very complex experimental design that's going to necessitate statistical tests that are different from what you would typically do. And so, I don't really think of there being, just to summarize, I don't really think of there being like a single SOP. There are some like guiding principles for how you would get started with your analysis, but invariably your analysis is going to diverge from the tutorials that we have.
Thank you. Yeah, that's a really good set of kind of guidelines or mental space to get into there first. Anyone want to add to that? Okay. Oh, Evan does. Hey, Evan.
Yeah, so if there's not like some SOP, how should I communicate what I've done with QIIME? Can I not just say I use QIIME to analyze the data? Do I have to like be more descriptive than that?
Indeed you do, Evan. Indeed. This is actually like one of the big things that sort of motivated data provenance, which you can see is very important to me. And so, when you're reporting your analysis, because it does a whole lot of sense to say, we did this analysis with QIIME or QIIME2, that's not going to be meaningful. That is not going to give somebody the information that they need to reproduce what you did. And so when I'm writing up a method section, you can find this in a lot of my papers. I'll have very detailed analysis steps, and sometimes that might look like the series of commands that I ran and maybe some annotation of what those commands were, and additional information like what versions of QIIME, what versions of reference databases I was using. And so that's really the information that you need. Now, one thing that I like to do is also put together of data artifacts and visualizations. And we did this for the QIIME2 paper that we published in Nature Biotechnology about a year ago. And if you see, there's a supplementary zip file in there that contains a few visualizations. And you can actually go download those visualizations and load them to view. And so then that way, somebody could actually review that and look at the provenance and see exactly what was done in every step of the analysis. And that is a really great way to document what you did with your QIIME workflow. And now there's one other thing that I want to mention here that's a really cool part of provenance. I'm not sure if we covered it yet. The citations also, the plugins will also have lists of citations that are associated with them. And so, for example, like in the analysis that we are doing in this workshop, if you were thinking about writing that up, you might think, okay, I better cite QIIME 2 because I use QIIME 2 for my analysis. But it's really important to remember that, like, not only did we use QIIME 2, we used, for example, SEP for doing the taxonomy, or sorry, for the phylogenetic reconstruction, and we used Data 2 for doing the denoising. So you use those, even though you use those via QIIME 2, but it's really important that you're citing those as well, because those are just really important citations for describing what you did and for making sure that everybody is getting proper credit for all the work that they did on all the software that they built. Anybody have anything they want to add to that? Okay, so Matt stepped away for just a second, so I am going to pull up another one of these questions. So one of the next question that I have here is, what would be the downside of using QIIME 2 to study extreme environments? Anybody have any thoughts on that one?
I can probably ping in with a little bit.
Yeah, go for it, Mike.
So, well, I guess the first definition is what do we mean by extreme environment? Because your skin, technically, for a microbe, is an extreme environment. So, you know, lots of salts and whatnot. I have a few co-authored publications where we looked at hypersaline meromictic lakes. So things that are very halo-tolerant microbes. We've looked at heat vents from leftover oil drilling holes, and we sampled these kind of man-made holes, but then they kind of were abandoned and they were naturally evolved through these kind of thermal vents. And we've had great success with using and studying these data. Now, this is where multiple issues might crop up, and that is the reference database you're trying to use to, say, classify these organisms. You probably want to use something that's more friendly for environmental data or cataloging. So Silva, in my case, since I've done a lot of these, is very beneficial. Certain databases are missing entire phyla from their reference database, like the candidate phyla radiation isn't even present or barely referenced in certain reference databases. So you want to use something that's kind of broad, or you can make your own. But generally speaking, in my experience, I have not had issues. Now, we could probably deviate into other sub problems like low biomass or deep 4 miles down below the Earth's surfaces, but there are people far more experienced than I on that field. But in terms of just being able to use QIIME to handle those data, too much of an issue. But if there's somebody else may have more specific points on that.
Thanks, Mike. Anyone want to add to that?
I think ultimately the actual data itself, you know, QIIME's pretty agnostic to that. It can handle whatever it can handle. It's just you know, the tools that you bring in or the reference databases that you bring in are what's going to be sort of the challenge, not so much, you know, the actual functionality of what's QIIME. QIIME doesn't really care about, you know, any of that. It's more just a set of tools or wrappers to allow the communication across these tools and platforms. So I think everything Mike said is spot on with regards to the actual data itself. I'm not sure if, you know, you would run into any issues in QIIME that you wouldn't run into the same issues using other sort of methods of analyzing the same data.
Thanks, yeah. I've got another ANCOM question in here. Maybe Jamie or Evan could take that. So the question is, how can I test the ANCOM assumption about fewer than 25% of your features changing between groups?
That's a really good question. Yeah, these are really hard to test without getting absolute abundances. So I think it really depends on what's your biological prior. So like I don't know. For instance, are you expecting there going to be massive microbial changes between your environments? And in the paper that we published, it's obvious in the sense that when you brush your teeth, yes, you do expect your entire microbial community to be perturbed. So this is one of those unfortunate scenarios where differential abundance is full of assumptions you can't really test. So my personal philosophy is try to reduce the number of assumptions that you make. But that's my personal opinion. Every differential abundance tool has their own recommendations.
Jamie, can I ask a follow-up?
Yeah, sure.
Like one way that I often think about that, because like you said, it is hard to test, is I think about like using ANCOM on environments that are more similar to each other. Like I might use it to compare like a gut microbiome between two subjects rather than like a gut microbiome and an oral microbiome. Is that a reasonable way to think about that? Like ANCOM is for more closely related or more similar environments rather than very dissimilar environments?
I think that's exactly the way you should think about it. Basically, the smaller your effect size, the more relevant ANCOM will be. So if you're trying to test completely different environments, then ANCOM might not give you the results that you want. Right, because the environments won't have any species in common. Now, if you test two different subjects, I guess it really depends how different the subjects are. So if the subjects also don't share any microbes in common, then maybe ANCOM might not be your best solution. But if you have a fairly small signal that is hard to tell otherwise, then ANCOM might actually be your solution to that sort of question. The old version of ANCOM, not the newer ones. It's very, very different tools. Mike has a question. Is that for this round or?
Yeah. So kind of to follow up this discussion about how to come up with ways to see if ANCOM is relevant, and I guess from the discussion, it sounds like we're talking about how much overlap we may or may not have between what we're comparing. So we're doing something as like a rough draft or rough pass to sanity check whether or not I should do ANCOM, to maybe do some sort of like core microbiome assessment or like a Venn diagram of sorts to see how many of my features above a certain threshold. I'm just trying to think of a practical way for users that might want to maybe get a little more confidence in their decision on whether or not to use ANCOM.
Yeah, I don't have a good answer for that. That's part of the unfortunate thing is that the assumptions are very difficult to test. Yeah, I'm sorry.
Could we base it off like a beta diversity metric? So if you saw, like, I am probably most familiar with the distribution in unifrac distance, but if you think about that sort of separation of the within versus between, and you start to think about that relative difference, maybe you can get it. My actual issue with ANCOM, which is related to this, is that I think that the ANCOM one built-in, so the ANCOM one built-in significantly different is based on this 25% assumption. And and that's where the 25% comes in. It's not actually the underlying method. It's the how do you get what's significant. And so if you're not sure that that's going to work out well for you, I might actually set a threshold a priori and say, I have 200 features. If my test is significantly different in 150 or more comparisons, then I'm going to say that that's differentially abundant, and that's going to be like my a priori threshold. And it doesn't entirely solve the 25% distribution, but it gets you away from the like bimodal significance assumption that I think is associated with that 25% thing.
I have some very mixed feelings about this. I think the takeaway from this is that one of the challenging parts of ANCOM is not entirely clear what the underlying assumptions are. I can come up with a scenario where where half the community is changing, and ANCOM will guess the wrong half of the community is changing. I honestly don't know. The ANCOM is a pretty difficult tool to wrestle, which is one of the reasons why I prefer the more recent differential abundance tools. I find them easier to interpret, and that also includes ANCOM BC, the latest iteration of this of ANCOM. But Justine, you brought up a really nice question about the beta diversity, and I want to kind of hammer this in because I think this is really important. That when you're doing differential abundance analysis, it doesn't give you the how significant it is overall. It won't tell you if there's a difference. It's just going to try to point out the most important microbes. If you cannot tweeze out a difference in your beta diversity, then you shouldn't expect your differential abundance to tweeze out a significant significance. You're running into the territory of p-value hacking, where you're trying to find a signal when there isn't one. So it's really, really, really important to make sure that that your beta diversity has a signal before you really dive deeper into your data.
Thank you. I'm looking at my queue here, and it looks like we're more or less through everything. We have just a few minutes left in today's stream. Jamie, I want to ask a question to you that is not related to differential abundance or ANCOM or anything else. We've been posing a question to all of our instructors here about what's your microbiome bioinformatics origin story? How did you get into this? How did you get into computing or into microbiome work or whatever?
My origin story. Yeah, my story is kind of weird. I started off actually interested in looking at microbial genomes and trying to do genome mining for antibiotics resistance genes. I joined Rob's group looking at microbial ecology with the intention of doing metagenomics. And I went into this really deep rabbit hole, compositional data analysis and microbe interactions and differential abundance. And I still haven't dug myself out of that hole yet.
That is fantastic. Thank you.
Do you think you'll ever dig yourself out?
No, I think I'm kind of addicted at this point. I'm hopeless.
Well, thank you for sharing that story with us and for joining us for questions today. I think I have one more instructor on the call maybe that I haven't asked that question to yet. Andrew, are you around? Hey, Andrew. Can I ask you to share your microbiome bioinformatics origin story?
Sure, yeah. I used to be a pre-med when I was an undergraduate student, and I got a lab tech job just because I needed a job. And it happened to be in an ecology lab, and they put me on a project that was using a computer. And it was an image analysis project in which they were trying to analyze growth just literally by counting the number of green pixels in pictures they were taking. And they were doing it in Photoshop. And they'd been doing it for years that way. And I discovered that Photoshop was not the tool to be using to do scientific image analysis in, and that their data from the past several years was actually completely whack. And so for me, that was some job security there. So I started researching ways to do it more accurately, and that's how I got into scientific computing. And actually, this was also at Northern Arizona University, and the one of the people in the lab there actually tuned me into Greg's lab, and this was like about 5 years ago, and that's actually, it's come full circle because I just joined Greg's lab earlier this year. And when I first got started, I actually basically just followed all the instructions on Greg's lab's website about how to learn how to program. And I pestered him several years ago about potentially working in his lab, but I don't think I was quite ready at that point. But yeah, so here I am, 5 years later, still without any sort of degree, but I have a job, and it's really fun, and I love it.
That's awesome. Thanks for sharing, and we're glad you joined the team. Let's see. I think I've got Anthony on the call as well. Anthony, could you share your bioinformatics, microbiome bioinformatics origin story with us?
Sure. So first of all, I guess I'm very much not as much of a bioinformatician, I guess, as everyone else here is. I'm really just a computer science major. Originally, I wanted to make video games and then decided not to do that. But by that point, I already knew I liked writing code, so I came to school for a computer science degree. And Chris Keefe, who's also here, already was working at Greg's lab and said, hey, you should apply and get a job writing code here. So I did, and now this is where I'm at, and I've learned just barely enough of the microbiome bioinformatics stuff to get by on developing QIIME 2, but I'm probably honestly know less about it than a lot of the students here, so I've been learning a lot from this workshop as well. Yeah, that's about it.
Well, thanks for sharing, and yeah, we're really glad you joined the team as well. I'll just mention out there really quick, the last couple of QIIME 2 releases have been significantly pushed forward by Anthony's contributions here. So he's been a huge help with moving forward all those giant code changes and integrating new features and dealing with bug fixes. So yeah, thank you. Is there anyone else on the call who I haven't had a chance to ask this question to? Looks like maybe not. Well, with that, I think we are just a minute or two over on the schedule here today, so we will wrap up. We will resume tomorrow and talk about longitudinal analyses and sample classification, and we're going to wrap up the Parkinson's mouse tutorial. So yeah, have a fantastic rest of your day, and we'll see you at the same time tomorrow.